All right, we should get going. It's 9:30. Welcome to day 2 of the AGM. Yesterday was a great first day to get us all going. I really enjoy listening to the projects that people have done and how they've applied the approaches from all of our work with GEN-FISH. So that's always a very exciting aspect of these types of meetings. Today, we're going to be going for more formal updates of the different activities in GEN-FISH and what we've accomplished. And then there'll be a roundtable discussion about where we're going or where the different activities can move forward into the future. And then we're going to wrap it all up with a very important legacy section. So I think we're going to be covering topics like potential commercialization and data storage and data management, which is going to be really important in the future. So I believe we're starting with an activity 1 update. So Margaret, take it away.
Are you pulling them up or I just have to advance and it'll be there? Oh, wow. Okay. I should have known that it would be well prepared. Yes. So we've heard bits and pieces of activity 1 discussions yesterday, particularly the several excellent talks from HQP yesterday. And so what I'll just be doing here is sort of a quick overview of activity 1, what we were planning to do and what we accomplished. So there were 3 key objectives for activity 1. So activity 1 is the eDNA fish survey toolkit. So the first objective was developing and doing that early stage validation of eDNA assays for all freshwater fishes of Canada, which tallied 218 species in total when you include extant native species and invasive species of concern. And so this is focusing on both the individual qPCR assays for each of the species at 2 genes, either individual qPCRs or the open array format that Chelsea was talking about yesterday, and then also optimizing metabarcoding assays specifically for Canadian freshwater fishes. The second objective was developing and validating environmental microsatellite DNA toolkit for quantification. And then the third one was, rather than using these tools for eDNA from water, but using it for stomach content to look at predator-prey interactions. And I'm just noticing how young we all look in those pictures, pre-GEN-FISH, pre-COVID. So just as sort of an overview, and I think that Chelsea touched on this yesterday as well. And so that the 3 main steps for developing and validating eDNA assays is first the sort of in silico analysis. So in silico, sort of computer-based, where comparing DNA sequences and designing primers and probes to have optimal match with the target and maximum mismatches with non-target species. So this depended on a really well-curated, comprehensive reference sequence database to be able to design those assays well. In vitro, sort of in glass or in tube validation, where we're testing those assays against tissue-derived DNA. And again, this depended on a really hard tissue collection to be able to do that thorough validation. And then in situ or in place validation. So the early stage initial in situ validation that was part of activity 1 involved testing the assays on water samples collected where we know the species occurred and where it didn't occur. Then the broad scale, large scale in situ validation is part of activity 2, part of specific case studies. So in terms of the in silico analysis, this was largely led by Stephen Rogers, a master's student in Bob Hanner's lab at the University of Guelph, where, as I was mentioning yesterday, he took 101 published CO1 assays and did a really extensive in silico comparison to predict whether or not they would amplify the target species as planned, but not amplify non-target species. And it was rather surprising and somewhat disappointing that only 5% of the assays were truly species specific, that they only ever amplified the target species, and about 20% were regionally specific, where they would work in some cases because the cross-amplifying non-targets didn't occur. And so Stephen worked out a decision tree where you could go through and, if it, blue was the ideal, where it was truly species specific, green was where it was regionally specific. In some cases, the red was where false positives were predicted, where co-occurring non-targets were predicted to amplify, or purple would be a false negative, where even the target species didn't appear to amplify, potentially because maybe the assays that were published used sequence from the species in a different part of the range, and it turned out that it probably wouldn't work on that species in Canada. So what we ended up doing then, this is one of the reasons of why I think Objective 1 took us a lot longer than we had planned. We were expecting initially to use a lot of published assays and then fill in the gaps. Because of the rather sort of underwhelming performance of the published assays, most of Activity 1, then we had to start from scratch to design our own assays, or in some cases, refine published assays. So this was a massive team effort. Largely with five labs coast to coast, from Scott Pavey's lab at UNB to Mark Shrimpton's at UNBC, where a lot of the initial work for this activity was then part of student projects, where students, largely working with regional partners, would have sort of a suite of species of interest, and then they would develop and go through the whole in vitro and in situ validation for the suite of species of interest to them. So just as a couple of examples, so Rena Cajoli at UNB, she developed or optimized Site B eDNA assays for 20 Atlantic Canada freshwater fishes. As we heard from Andrew yesterday, and Brittany's here as well, in Mark Shrimpton's lab, developing or optimizing or testing several assays for species of interest in BC. In Ontario, the Hanner and the Heath labs focused on a lot of species of conservation concern, or in many cases, particularly the Hanner lab, focusing on aquatic invasive species and developing and optimizing assays for those species. And in my own lab, I had a number of students, particularly Brooklyn, who joined us yesterday, and I'm not sure if she's on today, Jessie, Katrina, Morgan, and a whole sort of suite of excellent undergrads focusing on a number of Manitoba species of management concern or conservation concern. As we heard from Chelsea yesterday, though, that to really accelerate assay development and testing, we had to move to sort of the open array approach to be able to test sensitivity and specificity more en masse in a chip format, where 28 assays in duplicate could be arrayed on a chip for more efficient testing. So I've included a few of Chelsea's slides, but I won't, to sort of recap, but I won't go through all of the details. So this is the tally as of our last progress meeting, so that there's probably a few more that we can add to this, but we have 280 Co-1 assays for 183 species. So for a couple of key species, there's multiple working assays, and 191 Site B assays for 175 species. So still a few gaps, but doing very well. And we have tissue for in vitro validation for a large chunk of those. And that's what Chelsea has really been focusing on over the last few weeks. So we're in pretty good shape with that. And so the In vitro testing, as Chelsea indicated yesterday, involves looking at sensitivity with serial dilutions to see basically what's the lowest concentration that can be detected. And then also what was really key here is specificity, testing against non-targets, particularly more closely related non-targets, often within the same family or within the same taxonomic group, to test for cross-amplification. And so, as Chelsea was mentioning yesterday, so that there were 5 chips that were arranged phylogenetically. So, for example, the non-teleos chip, very conveniently, we had 14 non-teleos species, so that allowed us to put CO1 and site B on the same chip. So that would be the lampreys, gars, sturgeons, and bowfin, salmonid chip. And then there was also family Leuciscidae, so 28 species of native minnows. Another chip that had the other 28 other asteriophycins and a perciform chip. And then also, again, as Chelsea was mentioning yesterday, then there were also 9 regional open array chips that are being used for activity 2, but these chips could also be used with tissue-derived DNA for in vitro testing of specificity and sensitivity. And so again, just as an example here, the Labrador CO1 and site B chips, the diagonal is testing for the target species to see if the target species is amplifying. On the CO1 chip, there were 10 that didn't amplify, and on the site B chip, there were 11 targets that didn't amplify. But you might notice that the 10 species are the same, so that, as Daniel and Chelsea were mentioning yesterday, there's a good chance that it just might have been that the DNA for that one individual didn't work. So it's that those are not necessarily not amplifying the target. And then what was really promising is that there is relatively little non-target amplification. So the light green means that it didn't amplify where it shouldn't, and the blue is showing cross-amplification. Cross-amplification was just over 2% for the CO1 and just under 2% for the site B, which isn't bad at all. And in GEN-FISH 2, further refining, I'm sure will make those even better. So where we are then in terms of compiling all of this information to make sure that it's very detailed and ready for sharing publicly. So there's been a lot of effort by all of the HQP that are involved in the various assay development and testing, and then particularly with Paige, Chelsea, and Jesse, to really try to capture all of this information in at this point a Teams spreadsheet. So there's lots more rows, lots more columns where every piece of information that's critical for assay design and validation is captured, right down to the GenBank accession number for all of the sequences that are used, alignment files, primer probe sequences, numbers of mismatches, what species and what species it's been tested against, where those species came from, the number of individuals, just a massive effort to make sure that that's all captured. And this will eventually be shared publicly. Right now it's set up for sort of our within GEN-FISH use on Teams, but this is the sort of the basis for that eventual shared database. And then also, along with the validation, and I mentioned this briefly yesterday, that it took us quite a while to sort of work out a good, reasonably consistent, streamlined validation protocols. And now that those have been developed, a manuscript that's nearing completion now, largely led by the HQP, goes through some of these streamlined but rigorous validation processes with that sort of in silico, in vitro, in situ validation, and then outlining the important reporting requirements. And it nicely uses like 3 case studies, either how do you validate and optimize a published assay, how do you develop a de novo genus-specific assay, how do you develop a novel species-specific assay. And so it's largely meant to be useful for people who I won't necessarily say dabble in eDNA, but aren't necessarily hardcore eDNA experts. That there's a lot of resources already out there for the hardcore eDNA experts, but the idea is to and the whole idea of GEN-FISH is to put these tools more within the hands of the sort of the average biologist. And we'll be submitting this to a special issue of environmental biology of fishes that really focuses on knowledge co-production and actionable research. And just a plug, this is a special issue that is guest edited by a number of the GEN-FISH Fishcast team, including several of the HQP on the team. And the deadline is July 31st. If you think there's a paper that you have that would be of interest here. So in terms of, at this point in GEN-FISH, we're trying to get all of our deliverables and resources in a form that can be shared publicly. Eventually, a lot of this information will be on the GEN-FISH website, sort of joining some of the protocols that are already there, that are already being shared with partners to do with the field sampling protocols, DNA extraction protocols, and so on. And then there's a lot of other resources that we've developed for use within our groups that we also thought would be really useful to share publicly. So for example, all of our species lists that we have in searchable spreadsheets have really made an effort to use the updated classification that is outlined in the recent Mandrake et al. 2023 publication, and that goes along with the distribution, the sort of the native and the non-native distribution in Canada, so that the way the spreadsheet is set up, then it should be easily queryable or searchable by partners, where if somebody wants to generate a list of native species within their province, it's sort of a one click, or non-native species within their province. And so we're trying to organize and share those resources as well. In addition, again, stuff that we develop for our own use, we figure we might as well share. And so all of our spreadsheets, we've included information on the conservation status of the species, links to information. So again, somebody could easily generate a list of species of conservation concern within their province, or SARA listed species, or endangered species. And so it's all meant to be easily accessed. One thing I want to sort of mention briefly, then talking about Objective 1.1, is a project that wasn't specifically part of GEN-FISH, but sort of grew out of GEN-FISH and was funded by a side GLFC science transfer program project. Led largely by Chris Wilson and Paige and some other collaborators. And again, the idea with this is to really try to put these tools in the hands of users. In this case, really trying to help fisheries managers use eDNA, not necessarily to use it themselves in the lab doing the pipetting, but that they can work with eDNA service providers in a knowledgeable way to make sure that they understand what's being done and how they can interpret it. And so I will just very briefly go through what's here, but I really encourage you, if this is something you might be interested in, or some of your partners might be interested in, to take a look, because it's a really nice interactive system. 5 main steps. And so again, the idea isn't to reinvent the wheel. There's a lot of really detailed eDNA guidance documents that are out there, put together by DFO, or the Canadian Standards Association. But what we wanted to do was take the sort of the key information from some of these documents, but not too much that it would overwhelm the average user, and then put it into this interactive checklist. And so there's the 5 modules. If you enter, you can enter an email address or not, and then there's a series of questions, and depending on how you answer a question, if it's do you have a negative control? If you say no, then it will be a pop-up that will explain why a negative control is important, and some key but brief readings and so on to lead eDNA managers through it. Again, fisheries managers through it. Again, not because they're necessarily doing all the work themselves, but it also gives them an important list of questions to ask eDNA service providers, like, what is the specificity? What is the sensitivity? Has this been tested? Has that been tested? To allow for more dialogue between eDNA service providers and fisheries managers. And so, and what I really like about this is then, and I can say what I really like about it because Paige did all the work on this, so I'm not tooting my own horn. I'm giving credit where credit is due. That it generates a submission report. So if you put in your email address, that at the end you get emailed this lovely submission report that captures the answers to all of your questions. You can upload attachments. So if you have metadata associated with your project, you can upload your metadata file. If you have standard operating procedures with various steps. And so at the end, you have all of your various documents all collated into a single report. And then it sort of ends with a summary of how many yeses you had, how many no's you have, as an idea of how much additional work and discussion you might need to have. Next step, additional resources. So I think this is something, I hope this is something that will be sort of merged nicely with GEN-FISH to be another tool that our various partners and we ourselves can use. I suspect that my lab will be using this a lot, just as a way of collating all of our separate documents. So I'm not having to search through my files to find the metadata for this project or the SOP. It's all captured in that summary report that I received. So that was a quick-ish, considering the amount of work that was involved, overview of objective 1.1. And then I'll talk about the other two objectives as well. So the second one was the environmental microsatellite DNA development and quantification, largely led by Brian Neff at Western and Daniel at Windsor. And so this is where the idea was to use environmental microsatellite DNA to quantify the number of individuals. I'm sure most of you know that there's a lot of discussion about whether the relative signal strength or DNA copy number of eDNA can be used to estimate relative abundance. But the idea of the environmental microsatellite DNA is to actually quantify the number of individuals. And it's sort of comparable to allele counting used in forensics to try to determine the number of, say, contributors to a blood sample. That if you have 4 alleles, that you know that you have at least 2 individuals or at most 4 individuals, so that using that sort of allele counting approach to estimate the number of individuals contributing to the eDNA sample. So this work was largely done by 2 master's students, Simone at Western and Nabila at Windsor, as well as Dr. Sean Garner, research associate at Western, focusing on Atlantic salmon. So using eMDNA loci specifically developed or identified in Atlantic salmon, tested under 3 conditions, sort of starting in the lab where the known number of individuals in a tank, in stream enclosures, moving out to in situ validation in 3 Lake Ontario tributaries. And so again, just very, very briefly, but I encourage you to take a look at those theses if you're interested in more information. So with Simone's work, that going so that the sort of first pass of her study was to use a CO1 eDNA assay for Atlantic salmon to identify streams that had Atlantic salmon, and then use the eMDNA to try to quantify the number of individuals. She found that the microsatellite loci could be amplified from the water, but the genotypes of individual fish couldn't be reconstructed, potentially because of sequencing errors, working with minute amounts of eDNA in the water. One positive, additional positive that came out of this, though, was that these eMDNA markers could be used as a nuclear eDNA marker if desired, if there's situations where having complementary mitochondrial and eDNA markers would be beneficial. With Nabila's work, that again, looking at 3 Lake Ontario watersheds, and what was really cool with this is that Atlantic salmon quantification was done before stocking, 2 to 6 weeks after stocking, and then again 17 to 21 weeks after stocking. And she had pretty good success with genotyping getting genotypes from the samples, that 68% of the samples were successfully genotyped and assigned. We're able to assign an estimated number of individuals to each of the samples, again, using that sort of allele counting method. And as you'd expect or hope, that abundance was shown to increase after stocking in at least 2 of the 3 streams. And so one thing when we were talking yesterday about the how some of our work was received by the ROC and whether our initial proposal was overly ambitious, the eMDNA was one thing right from the beginning that when we were proposing this, there was like no confidence whatsoever that it would ever be possible. It certainly was difficult, but it has given some promising results that with further work, it may still deliver as promised. And then the last thing I'll just again quickly mention, and we heard talks from Jacob and Kyle yesterday talking about using stomach content DNA to look at either predator-prey interactions or trying to use stomach contents as a way of getting a fuller picture of the fish community in general. So there were sort of 2 steps to this, and this was largely what I'm describing here is largely Matt Sharon's master's project in Daniel's lab. And so initially, Matthew was testing using initially metabarcoding to identify stomach contents. And this was a large collaborative project with Ontario Ministry of Natural Resources dealing with 28 stomach contents from 2,800 fishes. And so initially trying to use metabarcoding turned out not to be as useful. We had a bit of a discussion about this yesterday about with metabarcoding, often what you get is a lot of the predator itself rather than the prey, which tends to swamp the output. Matthew tried using blocking primers that would block the predator sequence without blocking the prey, but there was too much similarity in the sequences that it was difficult to block predator without blocking prey. As an aside, that works very well in lampreys if you're trying to see what lampreys have been feeding on, because lampreys are sufficiently different from their prey, but when you're looking at more closely related Teleos species, it became more challenging. So this was, again, another good example of how GEN-FISH successfully pivoted during the course of the project. Initial plan was metabarcoding, but it turns out that using an open array chip, which wasn't in our original proposal, ended up working really well. And so having 28 species, potential prey species in duplicate arrayed onto an open array chip was really good for stomach content analysis. And a paper based on this has now been published in Molecular Ecology Resources. And then again, as sort of a unplanned offshoot of the original proposal, was using metabarcoding, even though it wasn't as necessarily as good as hoped for stomach content. This is key to activity 2, the 500 water body project. The original plan was to use existing metabarcoding primers to do all of activity 2, but there's been a lot of effort to improve some of those primers for specifically for Canadian freshwater fishes. So a lot of this work, and there'll be more that you'll hear about in activity 2, has been a large focus of work in the Heath Lab, largely led by Matthew Yates. And so I'll talk first about one of the reason for wanting to improve metabarcoding for Canadian freshwater fishes. And so this is some work that came out of Bob Hanner's lab, largely led by Erica Myler as part of her master's thesis, where she looked at taxonomic blind spots that resulted using existing primers, finding that so that she tested existing metabarcoding primers against known communities and compared to fish composition with conventional sampling, and saw a lot of blind spots, a lot of species that were missed with the existing metabarcoding primers. And these aren't atypical that we're sort of aware of it. First of all, universal primers are rarely truly universal. They sometimes entirely miss species or miss taxa, or at the very least don't amplify them in the same proportion as they're represented in the fish community. There were some other challenges because of gaps in the reference sequence libraries. It's one thing to get a sequence, but then if you don't have a species to match it to, that can be problematic. And then also with small fragments that are used in a lot of metabarcoding studies, often it doesn't allow for sufficient resolution between closely related species. And so that's where the sort of new and improved metabarcoding primers come in. And so in the Heath lab, there was a lot of effort to explore other primer options and also try to amplify a larger amplicon, with the sort of expectation that if you have a larger amount of sequence, there's a greater chance of resolving, particularly among closely related species. And so this is a manuscript that's in prep, and again, another one that I think is close to being, very close to being submitted, where trying to improve on the existing 12S metabarcoding primers has led to improved resolution for Canadian freshwater fishes. And I think this is my last slide, and it'll lead, I think, nicely into activity 2, where just to sort of summarize some of the key findings in this paper. Again, if the new and improved 12S primers are compared to the existing ones, out of the 173 Canadian freshwater fishes that were in the database, 167 were identified with the new primers compared to 162 or 160. And then again, this is going to lead very nicely into activity 2 and the case studies then. Thanks.
Nick, if you're online, I believe that's your cue.
Okay, here I am. I wasn't sure if you just wanted me to get right into it. Okay. Thank you, Margaret, for that great overview. And you're right, that leads right into activity 2. Matthew Yates and I are going to tag team this. So in a couple of slides, I'll just ask Matthew to jump in and talk about the 500 water body survey, and then we'll tag team a couple of times after that. So the 2 main objectives of activity 2 were to do this broad scale spatial temporal eDNA assessment of fish communities in 500 water bodies. And then the second one was to do end user identified case studies on the application of eDNA tools for population level assessment. And so we will not get into each case study in detail. I cherry-picked a couple where there's some recent results or updates that you probably have not heard at previous AGMs or through my presentation or through the presentation of HQP. Next slide, please. So within objective 2.1, this is the sampling of the 500 water body for eDNA with collaborators and end users. We talked a bit about that yesterday. And then applying quantitative PCR and metabarcoding methods to determine fish community composition. And Margaret went over some advances on that front. And then the part that we just ran out of time with, and I look forward to having a discussion about the future and hopefully the near future, about task 2.1.3, compare historical and eDNA fish community data and use biotic and abiotic data to assess environmental effects on eDNA detection, efficacy, and reliability. So, we're just about at the point now where we have a data set for which we can do this, but we've run out of time. So again, when we talk about the future, hopefully the near future, we'd like to include this task in the discussion. Next slide, please. And at this point, I'm going to hand it over to Matthew because he really quartered back the 500 water body survey.
I'm assuming this is a pretty standard rig. Okay, sweet. All right. So basically, yeah, I was the coordinator for the activity 2 500 water body survey, which was a pretty immense pan-Canadian effort that relied on a lot of collaboration with government, academic, and community organizations to collect samples distributed from across Canada. This is actually a slightly outdated map. We actually have more samples that are presented on this map, but we'll get into that in the future. But you can see that basically we spent a lot of early years just sort of getting as much as we could, and then the later years sort of filling in the geographic gaps, particularly for certain different biogeographical regions in Canada, particularly the north. So let's get into it. So yeah, as I mentioned before, it was a pan-Canadian effort from tons of different agencies and groups across Canada. We were successful in collecting samples from 9 out of 10 provinces and 3 of the territories. Unfortunately, we didn't get anything from PEI, but it's a pretty small province. This was all metabarcoded in-house on using the long amplicon metabarcoding primers that we designed through GEN-FISH that Margaret discussed shortly ago. Most of this DNA was extracted as in-house as well, although some eDNA was donated to us from different organizations, particularly the provincial and federal agencies who have their own protocols for handling that. So when the dust settled, by the end of the program, we collected 3,887 samples from across Canada. That's almost 5,000, including the different controls included as well, which all have to be analyzed. From 1,400 different sites, so unique geospatial locations present in Canada, from 586 unique water bodies. So it was a pretty immense effort, and in terms of the number of samples collected, we actually were able to overshoot the targets, which was an already ambitious 500 water bodies. In terms of sequencing, we're a little bit far behind. Everything's been extracted at this point, but the number of samples we have sequenced so far is 2,371 from 1,115 sites from 445 unique water bodies. You'll note here that the ratio of the number of sites over the total sites that we collected from and the number of unique water bodies over the total number of unique water bodies that we collected them from is higher than the ratio of the number of samples relative to the total. That was intentional because we tended to favor, we had some projects that had very intensive sampling of certain water bodies, but when we were sort of generating the data set, we tended to favor getting broad geographical representation in the data set over repeatedly sampling water bodies. But this will all be sequenced. We're on track to do that by the end of June. The data grows daily. In total, in terms of the plate by marker combination sequence, we have 117 plates sequenced for a given marker or with a metabarcoding marker in total that have led to 9,000 unique species detection events. We've generated 63 million fish sequence reads, and in total have sequenced 16 billion base pairs as a part of this project. So it ended up being a pretty immense effort, and it's, again, we're about 2/3 to 3/4 of the way through it or so. But this is pretty fresh off the presses. We were finally able to sort of wrangle our metadata into a format that we can actually start analyzing. And so this is a broad map of what we've accomplished in terms of sequencing and the number of species detected at a given spatial location. And you can see already that there are biodiversity hotspots that are jumping out already, the key one being southern Ontario and the south of Manitoba there. But we have pretty solid representation of sequencing from across the country already. already. Again, that was intentional because, you know, we have pretty intensively sampled data from Newfoundland and Labrador, for example, that was donated very generously from DFO, but we had to make sure that we were sequencing samples from across the country as well. But we're now returning to those samples to finish off on those. We have a little bit of the main sequencing effort we have to remain is a bit in the north in Wapusk National Park and some samples from Nunavut. There's a few in the southern Manitoba area that we need to get finished, and of course the high density sampling in Newfoundland and Labrador. But that's coming along. So that's just species richness, though, looking at biodiversity hotspots. We can actually start asking, well, how does fish community composition differ, at least according to the eDNA samples we collected? Now, as Margaret alluded to, there's a lot of debate about whether or not you can use eDNA as a metric of relative abundance. I am solidly in the favor of the camp that you can. So we started looking at relative breed proportion in our metrics of community composition. And we did that. I did that by basically calculating what's referred to as an eDNA index value for each species present in a community. That just uses what's called a Wisconsin double standardization, and it's kind of recognized as the best method to sort of quantitatively interpret metabarcoding DNA. And then I use those values to calculate a Bray-Curtis dissimilarity distances for all of the different water bodies present in the survey. And again, this uses the relative abundance of the reads. And then I put that output into an NMDS analysis to compare community composition in water bodies across Canada. So this is how it looks. And this is really hot off the presses. I was doing this yesterday. So we're kind of really sort of just starting the analysis of this data. So you can see here that this is grouped by province. So it's pretty messy, but you can definitely see some clusters starting to form. But obviously, it is probably going to be messy because provinces are defined by relatively arbitrary imaginary lines that the government decides to draw over land. And so there's a lot of different sort of biogeographical components and spread across all of the different provinces. You can see Manitoba, for example, has a pretty wide range of habitats, particularly compared, at least wide range of community compositions compared to a lot of different provinces, because it has the very diverse Winnipeg, boreal forest, and then you get into the tundra at the beginning. So it's solidly kind of in the center there. But you can see sort of the East Coast provinces clustering out, the West Coast and the Northern Territories clustering out a bit as well. But what happens if we group it by region? Well, there we can really start to kind of see clustering pop out. So I define this as basically the Atlantic region, central in terms of Quebec, northern or Quebec and Northern Ontario, the Great Lakes region, Newfoundland and Labrador, the Northern Territories, the Prairies, and then the West Coast. And yeah, you can see that we pretty clearly start observing clusters. Now, I have the ellipses in there to sort of as an estimate of the variance around community composition, but I find they are sometimes not helpful for visualizing the graph. So when you get rid of those ellipses, you can actually really start to see the clusters emerging from the data. Now, one thing about NMDS analyses, though, is that the distances that are calculated and sort of the composite differences amongst the communities and the ordination, the axes, the orientation of the axes are relatively arbitrary. There's not the positive or negative numerical values of those don't have any sort of intrinsic meaning. So what we can do is we can actually sort of tilt this and flip it. And then when you look at this and you look at how the colors correspond to when we flip it so that the secondary axis is on the x-axis and the primary axis is on the y-axis, so sort of invert the figure. When you look at it, you can actually start to see the regions sort of popping out in the traditional arbitrary orientation at which we usually look at Canada, the north, south, east, west. You can see we have Newfoundland and Labrador here on the northeast with Atlantic Canada just underneath it. We have the southern Great Lakes clustering out at the bottom, central Canada, brown in the middle there, the prairies just to the left or west of that, the Northern Territories on the north clustering out with Newfoundland and Labrador, very closely related to as well, and then the West Coast oriented there. And if you kind of squint real hard and squish a map of Canada onto it, you can actually basically see the geographic spread. Now, I want to emphasize here that there is actually zero geographical input or data inputted into this analysis. This analysis is composed entirely of just community composition data. So when we're looking at the distances estimated in the sort of Bray-Curtis and MDS analysis between communities in Canada, they naturally form a biogeographical relationship. So you can see that MSDS axis 2 roughly corresponds to an east-west gradient, and the primary axis corresponds to roughly a north-south gradient. So I was pretty pleased when this figure popped out of the data. I was pretty excited to see that. So, I mean, this is kind of just the beginning as well for this type of analysis. These are just basic biodiversity analyses and community composition analyses. This data set is an incredibly rich one. There is so much that we can mine from this data set and use to analyze and answer interesting questions in the sort of in ecology and in the study of environmental DNA. So in terms of what's next, obviously finishing sequencing is important. We're going to finish that hopefully by the end of June, the end of GEN-FISH. We're on track to do that. We only have about 15 plates left. The majority of those are already library prepped or are in progress. And I think we've got about 5 more to do in the last 2 weeks of June, then throw on the sequencer, and then we should have basically everything done. But once that's done, we can do a lot of different things. One thing that I'm really interested in, and we touched on this a lot yesterday, is haplotype diversity. Like, how can we use eDNA to sort of look at and examine haplotype diversity across populations, or intraspecific diversity across populations within Canada? And so that's something that I think we're going to try to take a solid look at using this data set. But again, analyzing things like the biogeography of fish communities, drivers of biodiversity in Canada, which pretty much turns out to be an east-west, northwest gradient. The things like eDNA sampling effort needed to quantify biodiversity, and the things like how much benefit do we get from adding additional markers. I also want to emphasize as well that this is based off of a single marker on uncleaned data. This is actually data that has not been cleaned for potential contamination, which you can do based off of analyses that you do on your negative controls and your field controls. So there are actually fish popping up in some of these locations that shouldn't be there, that we will clean up once we sort of analyze our controls. And this is only going to get better as we clean the data and add 2 more markers to it as well. So, and I want to also emphasize too that, again, as Daniel was mentioning yesterday, the scale of this is really kind of unprecedented. You know, like, I've done a lot of work looking at, for example, the sampling effort needed to quantify biodiversity on a small water body scale. And there's been a lot of work in the eDNA literature to do that. But we can actually take this data set and we can start looking at, okay, when you have a very large area with multiple different biogeographical regions that have different ecological characteristics, how is the best way to go about conducting those types of analyses? And that's unprecedented in the literature. And that's something that I'm hopeful that we're going to be able to accomplish using this analysis. You know, like, how do we better inform sort of really large scale studies of biodiversity using these tools? And so, yeah, I mean, the data set is going to be pretty much generated by the end of June. I'll be spending the summer cleaning it up, but, you know, there's the, you know, your imagination is the only limitation when it comes to this data set, you know, and so this is a data set that's going to be open for collaboration for anybody in GEN-FISH, you know, or really anybody ultimately, once it's made public. And so, you know, there's an open invitation for collaboration here. So if you guys have any questions that you think you might be interested in addressing using this data set, feel free to send me an email and we'll chat about it. So, yeah, I also want to send a huge shout out to the GUIR team that basically made this possible. Raj and Ahmed were you know, foundational in the lab doing all the PCRs and extractions. Chelsea Frank and Kyle Banwell also played key roles in doing metabarcoding and eDNA extractions for this project. Jonathan LeBlanc was involved in sort of getting it off the ground and really key player in that. And of course, Shelby Mackey, who does all of our sequencing. And again, I'd like to thank Nick, who was the activity lead for this, and Daniel, of course, who was the one who made the whole thing possible as the main lead of GEN-FISH. And of course, again, I want to mention that this was only made possible by a collaborative effort of labs across Canada, of which many GEN-FISH labs were major players and donors. So essentially, most of the samples represented in this data set were donated by labs that were working in as a part of the GEN-FISH project. So it wouldn't have been possible without all of your guys' contributions as well. So with that, I'll pass it back to Nick, and then I'll talk a little bit more later on about a case study.
Okay, thanks, Matthew. Yeah, you did sort of dig into our future there, but so people should remember that for later today when we talk more about what to do in the future. And I see Anne Lurie has some really nice comments in the chat about some of this work, and I look forward to more discussions about those comments during the futures talk. Next slide, please. So we started off with 8 case studies. We added another 9, and we ended in across those case studies, we had 14 partners and co-producers. So I think this is a testament to the interest in GEN-FISH, and once we started talking to partners about the questions that could be answered, potentially answered, or to determine whether or not questions could be potentially answered with eDNA, that really got people thinking about additional applications that led to these additional case studies. And you can see that there's a wide variety of topics that were covered in these case studies, from distribution to movement patterns to biodiversity, diet, and conventional versus environmental DNA sampling. Next slide, please. So here are the original case studies, and as we had presented them to Genome Canada in our application. And you've heard about many of these case studies, the results of these case studies in the past. Again, I'm just going to talk about a couple of them in just a minute. Next slide, please.
Sorry, it's slow.
Okay, no worries. It is very slow. There we go. And so you can see that we added additional case studies. These are the additional ones with a wide variety of collaborators and a wide variety of topics. Next slide, please. Well, we went. This is actually case study number 8. Matt Sharon did some of that early diet work under activity 1. We did have a question from Saugeen Ojibwe Nation about whether or not stocked lake trout eat the lake whitefish that they fished. And there was several false starts in my lab with not being able to find the right PhD student to do this. And within this last year, we have had a postdoctoral fellow, Emma Lemberg, work on this question. And we have 150 lake trout stomachs collected by M&R around the year 2000, and then they have identified the contents morphologically as fish to species, unidentified fish, inverts to species, and unidentified inverts, and then separate jar for just the chyme of an unidentifiable mush. And so Emma is working on identifying those contents so we can do a comparison of using 3 different methods. So we can do a comparison of qPCR based in just for lake whitefish versus metabarcoding for fishes only versus use of the open array chip, and then versus the actual morphological identification. So I think this is a project that nicely complements Matt Sharon's work and Jacob and Brian's work to really, you know, in the end, we've really done a deep dive into looking at the use of stomach content DNA in GEN-FISH. Next slide, please. And just a quick schematic of what Emma's doing. Her samples are about to be sent for sequencing now, and we expect that she will work up the results by the end of summer. I just want to point out one thing that was problematic with Matt's work, which was the prevalence of the predator's DNA in the stomach contents. Well, one of the ROC meetings, someone suggested that we use a peptide nucleic acid clamp to exclude the predator DNA, and Emma has designed one of those clamps and is using it, so we'll be able to evaluate the effectiveness of that clamp as well. Next slide, please. Just briefly, Markel's whole PhD is centered around GEN-FISH case studies, and the one that I don't think she's talked about yet is this determining the abundance of spawning lake trout and associated egg predators using eDNA. And this is where she's Saugeen Ojibwe Nation is interested in finding better methods to identify the distribution and timing of lake trout spawn, lake whitefish spawning in Lake Huron, so that they don't have to go out and manually sample in order to determine whether the fish are spawning. So Markel has been out there in November. This is a very difficult time to sample. It's quite rough, and there's a lot of cancellations due to weather. And I just wanted to show some preliminary results. So what she does is she basically, before they set the net on the spawning shoal, she takes an eDNA samples. Then she sets the net. They set the net. They come back the next day. She takes eDNA samples. They pull the net. And what you can see on this graph here is the mean eDNA quantity, copies per liter, and the biomass that's caught in the net over time. And as the biomass increases, that means that they're going into spawning. And you can see that there appears to be a relationship here based on 2 years of data for lake whitefish, and actually a stronger relationship for Cisco. And we're now looking for funding for Markel to go for a 3rd season in order to decrease the sample size a little bit this November. Next slide, please. Now I'm going to send it back to Matthew to talk about the final results from this interesting project involving goldfish in urban stormwater ponds.
Yeah, so this was thanks, Nick. So this was a case study that was developed in collaboration with DFO. They were really interested in kind of examining the utility of eDNA to monitor invasive species in urban environments, as well as quantify fish community biodiversity. So what some smart folks realized was that stormwater ponds actually represent very ideal study systems to examine these types of questions, because you can drain them. And so that's something that's very difficult to do in natural ecosystems, draining them allows you to get total census estimates of fish present in these ponds. And that's something that's extremely difficult to do in natural ecosystems, obviously, because if you drain it, you're going to destroy it and kill all of the fish. So, so this is kind of a unique opportunity to actually generate validation data for eDNA or generate eDNA data and compare it to validation data that has a very low amount of error in terms of determining community composition. Because things like electrofishing, seining, all of those come with a certain level of error. They're not necessarily foolproof techniques themselves, but draining a pond and counting the fish that are flopping around on the bottom of it is a pretty good way of actually counting what exactly is there. So the goal of this was basically to compare genetic and conventional census estimation methods for goldfish, to basically use eDNA to not only detect them, but also compare the detection relationship between their detectability and abundance and the concentration of eDNA that we observe. And then also we did community biodiversity work, which I'll talk about in some later slides. But one thing that we did here, what a major goal of the study was to examine how replicable are these findings across different laboratories. And so we basically collected samples. We collected 3 samples from a site. We kept one for safety. GLEAR took one, and then DFO took one. And we all extracted the samples using the same exact protocol and then analyzed the concentrations of eDNA from those samples collected at the same sites using the same qPCR assay and techniques. And we ended up finding that there was an extremely high replicability between site-level eDNA concentrations estimated by the research group here at GLEAR and the research group at DFO. Once you exclude those 2 major outliers there, which kind of skew the curve a little bit, they were extremely high concentration samples. I don't know, we must have got like a piece of a goldfish on the filter or something like that. But once you correct for those outliers, the marginal R² between the relationship between the 2 assessed concentrations at a sample level was 0.945. So basically the concentrations from one group explain 95% of the variation in the observed in the concentrations estimated by the other group. So that's an extremely highly replicable relationship. When we actually take those concentrations from a specific pond and average them, and then compare the average concentration for the pond, which is really the unit that we're interested in if we're trying to estimate the abundance present in a pond, we observe an even higher or correspondence between those estimates with a marginal R² value of 0.93 or 0.993 in the average pond concentration estimated by each working group. So I've worked in science for quite a bit now. I've reached the point where I have gray hair, possibly in part of working on it. But I've never gotten an R² correlation that strong in my entire life in data that I've generated from natural ecosystems. So I think this just means it's further proof that it is a very highly consistent and replicable tool and means for assessing biodiversity and species presence and absence. So very, very consistent relationship across working groups. And again, because this relationship was so consistent across working groups, we again found a very consistent positive relationship between goldfish abundance and the concentration of eDNA that we observed in these ponds. Interestingly enough, goldfish biomass was a pretty terrible relationship, didn't correlate very well at all. But the abundance, the numerical abundance, correlated quite well, as did and allocating or accounting for some metabolic processes that we hypothesize are involved in eDNA production also further improve that relationship. But naturally, the relationship between abundance and biomass was pretty good, or pretty good. So it's probably a pretty reasonable technique to use to assess goldfish abundance, at least in these environments. And we again see the same relationships observing when we look at the detectability. So this slide was created from census estimates obtained from draining the ponds. But you can use conventional estimates like catch per unit effort from seining or electrofishing to generate estimates of abundance as well in these ecosystems. And so we also DFO did a ton of conventional work on these ponds. It was a huge effort. And we compared the detectability of goldfish eDNA relative to the the sort of abundance estimated from catch per unit effort metrics from seining and electrofishing. And again, we see the same relationship. There is an extremely strong positive relationship between electrofishing catch per unit effort and seine catch per unit effort and the detectability of goldfish. And in fact, once you get above a certain threshold, we can basically estimate the threshold, catch per unit effort metric above which you only need a single sample to reliably detect goldfish in these ponds. So again, and that's a pretty low level of effort. It occurs somewhere around 0.08 fish per minute. So you can electrofish for almost 10 minutes, find a single goldfish, and basically you know that you're going to be reliably able to detect that goldfish from a single water sample that you collect from that pond. So again, highly sensitive technique. And and again, this type of work is the type of work that we focused on doing with our collaborators, because it's really applied stuff. Like, there's not a ton of theory involved in this, but this is the type of work that organizations like DFO want to undertake to validate and start seeing how reliable these tools and techniques are, so that they can sort of start integrating them into their toolkits and develop sampling guidelines for their use to monitor invasive species and quantify community biodiversity. So in addition to doing qPCR on these samples, we also metabarcoded them as well. And so the focus of this was not on the goldfish, that was the qPCR study. This was on this part of the study was focused on quantifying community biodiversity. So we compared the effect of different markers on detection probability and ended up finding that some markers work better than others. But as part of that, we also determined the minimum number of samples needed to quantify 95% of the fish biodiversity present in a pond. And the only thing that we ended up finding that affected that was actually pond size, which is kind of unsurprising. The larger the pond is, the more samples you need to take to make sure that you get all of the DNA from all of the fish present in that pond. But again, this is really practical sampling guidance or guidelines that we can generate in collaboration with our partners. We can basically say, based on the size of a pond, an urban stormwater or an urban pond, you need to collect X number of samples to reliably quantify fish community biodiversity. And again, we also did other things like looked at the effect of sample location on the number of species recovered. Didn't matter. It seemed to be pretty arbitrary across ponds. But we also, again, compared the reliability of eDNA for quantifying community biodiversity relative to conventional sampling like seining and electrofishing. And we did this knowing that we had total community census estimates from drainage. So we could look at the reliability of all the different methods. Now, we ended up finding that eDNA pretty reliably detected more species, but reliable is not necessarily the best word there, because a lot of that was driven by false positives. So detection of species that weren't present there. So this again highlights the need for stringent laboratory and field protocols to control potential contamination. But overall, it tended to work quite well. And again, we ended up demonstrating that there seemed to be a moderate correlation between the eDNA read count and the relative abundance of species present in these ponds. And this was a consistent relationship across the different markers. Some markers performed better for estimating relative abundance compared to others. Notably, the GEN-FISH design markers actually performed quite well for predicting relative abundance of fish species in these ponds relative or based off of relative read count. But one thing that kind of came out of this that I thought was a really interesting finding is that we actually can use eDNA, our eDNA data, to infer and inform what conventional methods are more reliable. Because we ended up finding across every single marker that the relative read abundance generated from eDNA data exhibited a much stronger correlation with data produced by seining relative to electrofishing. And the only way that's going to emerge is if seining, the only way I can think of, unless there's some weird biases that I'm not not capturing. But the only way I can really think of or explanation I can think of for why that is occurring is that seining more reliably reflects fish abundance relative to electrofishing. So we were even able to generate data to inform on the effectiveness of conventional sampling as a part of this project. So I'll hand it back to Nick for that. And yeah.
Okay, just the last slide. Thank you, Matthew. So towards the beginning of activity 2, we wrote a paper on the applications of eDNA in freshwater fisheries management and conservation in Canada, where we did an overview of current challenges and opportunities. Well, now we're going to bookend that paper with a paper that it compares some of the end user case studies, and we hope to submit that to the special issue of CJ-FAS in the very near future. It looks like most of the case studies write-ups are completed, and now it's back to me to compile and smooth out the final manuscript. And that's it. Thank you.
Before, Amor said, wondering if you say the zones or equal provinces of Canada could be. He also shared a link to terrestrial ecozones and.
Yeah, definitely. That was like a super. I literally was doing that last night when I got doing those analyses. But definitely, I do find more fine scale sort of biogeographical analysis would be the next step. Those were pretty quick and dirty blocking space of region to conduct that analysis. So yeah, I think there's a lot of information there, which is pretty cool. Sorry. Yeah.
Didn't go too well yesterday. Okay, just a few more comments in the chat. Nick Mandrack had said, good idea. There's also Kasiwik's National Freshwater Biogeographic Zones. Anne Lore said, quite an amount of work indeed. PC is really interested by large-scale approach for monitoring biodiversity. And she also said, she's very much looking forward to seeing publications associated with these case studies and wondering if there would be any. Celine says, very convincing outputs from activity 2. Great.
Just a quick question, and that's knowing that there can be regional specificity. When you're going to be mining the data set, 500 water bodies, will that play a role in the kind of conclusions or interpretations of the data that you can make? Given if there are regional differences, what kind of level of confidence do you have, unless you'll be, or are you applying the sequences at a regional scale to the 500 water bodies too?
So the 500 water body data was generated from metabarcoding, which is a bit different from the qPCR and the use of universal primers. So the likelihood of there being mismatches at a regional level and the binding sites of the universal primers, which tend to be extremely conserved, is a lot lower than the sort of intraspecific variation you observe that can be challenging when you're trying to design species-specific, universally species-specific qPCR assays. So it's much less of a problem. I don't anticipate it really being much of a problem at all. What we can do, and we actually take, the folks at TIPO might potentially do this because they did this with a case study. I think might help us out with the sort of broader cross-Canada comparison, but you can do in silico analyses to see what the projected amplification efficiency of the markers we used are species. And so all you have to do is make sure that you have sort of representation from different regions in Canada to see if any of those pop up in silico analyses. I don't anticipate that to be a very large problem though, for at least with that market.
Thanks.
And Matthew, your analysis of haplotype diversity could be used as a warning.
Yes, yes,
So if you find a species that has a lot of haplotype diversity,
yeah,
then you may be missing some,
yeah,
if you see what I mean.
yeah, definitely, not for humans. That's-
What's a false positive electrofishing?
That is a very good question. It's great that you picked up on that. So that actually reinforced, this is something we discussed in the detail. But basically what ended up happening is we would go in, we would collect the eDNA, then DFO would do the conventional sampling, and then the pond would get drained after that. Now, how long that pond got drained after the eDNA and conventional sampling was done varied. There was up to 6 month difference in one case, but predominantly it was done within the next month. Sometimes immediately afterwards, but there is that temporal gap. And so an electrofishing false positive occurs when you electrofish, you catch a species, but then it's not present there when you drain the pond. And these ponds are semi-open during high water events, so fish can move in and out of them. What's really cool about that though is that so there were some contamination challenges with some of the DFO work. We think that might have to relate to how they do their PCR processes, because they're really interested in rare species of conservation concerns. So they use more PCR cycles, which can potentially lead to more false positives, because they are far more sensitive to false negatives than false positives. Like if there's a rare species in there, they want to know it. So their protocol tended to produce more false positives than the one that we used in Clear. When you looked at those graphs, the false positive rate between Clear and electrofishing is not statistically different. So the fact that electrofishing has a false positive rate implies that there's going to be a natural background false positive rate for eDNA as well, because you're going to detect species using eDNA that were there, but were transient, no longer there when the pond was drained. So that's actually even more robust evidence that if you're careful about your protocols in terms of, if you're worried about false positives anyways, and how DFO does their eDNA work, it's very intentionally designed, so they know that there's a trade-off in terms of what they do in generating false positives versus false negatives. But if you're worried about false positives, there are things that you can do to mitigate that, and the data that you generate is comparable in terms of positive rate that we might get from conventional sampling.
But did eDNA detect that electrofish wanderer?
No, it didn't. Both eDNA
Did not detect, what was it?
it was a white sucker.
Okay, and it didn't show up in either eDNA?
Yeah, it didn't show up in eDNA or or the training either. So there was a difference there. You didn't pick it up with them. Yeah, but they caught it fishing in. So
I think we'll have to, if we can put a pin in the questions, so that you and Ken can answer your second.
We want to get to the important stuff.
Lunch.
Lunch.
I can change.
Yes.
Okay, good. All right. So I get to update you all on activity 3, which was the development of the fish health toolkit. And just as a sort of a reminder of what was involved in the development of the toolkit, it was to produce one of what we were calling the stress response transcriptional profiling chips. This initially started out as a goal of having a universal qPCR assay for all the Canadian freshwater fishes for 112 genes, which I think we've said now many times over the last couple days was fairly ambitious. I would argue impossible in this instance. And so what we've ended up doing is in our attempts to produce these 112 assays, we ended up producing a large database of assays that we could then use to design some of these 56 gene chips and these 28 gene chips. And just the reminder of the process, it was essentially we were either producing our own transcriptomic, our own reference transcriptomes for our species of interest, or we were downloading them off of public repositories, aligning the sequences, and then putting the primers and probes, designing those in the conserved regions between all the different species. And so within that effort, we were able to produce a lot of these smaller STP chips. As I was talking about, these were largely designed to work for gill, muscle, and liver. We also designed ones that were a little bit more specific to brain. And then sort of an add-on component to the project that was not part of the original plan was to develop this mucus chip as well that we heard about yesterday. And so when it was all said and done, we now have STP chips that work for salmonids, for acipens or sturgeons, for lampreys, cyprinids, perciforms, and then also there is one designed for the American eel. And so just similar to what Nick had mentioned, we'll just be sort of talking about either sort of the early development of the chips and how they were being tested, or sort of the more recent developments that haven't been talked about at previous AGMs. So early on, Shahin Islam, he did a lot of work producing that 112 gene chip for salmonids. And then this was tested on liver, gill, and muscle from 8 different species from 4 genera of salmonids. So it's pretty, the goal was to see how well these conserved assays would work across different species. And he was able, with that approach, to actually see some amplification in the 3 different tissues across the 8 different species, which was pretty impressive. It was a little bit more of a challenge to get them to work well with the whitefishes, but it worked quite well with the Oncorhynchus, Salmo, and Salvelinus species. And so from that work, then it was sort of the big proof of concept, and we were able to then really be sure that our smaller chips are going to work. And again, producing that database of assays that we could then make these other specific chips that would be more relevant for either the specific question for that study or for a specific type of stressor. We also developed this acipens or sturgeons chip. This is a 56 gene chip that was sort of focused on thermal stress, general stress, and food deprivation. This was designed by Kyle Madden in Nick Bernier's lab at Guelph, and also Hossein Hajiji in my lab at the University of Manitoba. And so they produced, again, following the same approach, grabbing all of these sequences from as many different acipens or sturgeons as we could get. And then these were then tested on 4 different species of sturgeon that are relevant in Canada. We've been trying to get green sturgeon samples from California. It's just they're a protected species, and that becomes a bit of a challenge. And so these were then tested on the liver and gill for these 4 species to make sure that the assays were working. And then the chip was designed and it was used for some of the master's thesis work for Kyle, and then also for one of the PhD chapters for Hossein. For Hossein's work, he tested these how well the chip performed when looking at fish that underwent an acute thermal challenge. So this is going up to a CT max for 3 different species of acipens or sturgeon, and a CT max is just when you've acutely heated a fish to the point that they basically lose equilibrium and can no longer function. And so since then, we've now refined this chip, and a version 2 has been printed and has been tested. So it's evolving, and it's been I'm pretty excited that we were able to get one to work for acipens or sturgeons, because many of them are octoploids, which makes it a lot more complicated than you'd want it to be. And then again, this was a situation that was not part of the original proposal, but it was sort of suggested by partners as a species of interest for the STP chip technology. So Patricia, we heard from yesterday, producing chips for cyprinids and perciforms. Also, in your spare time outside of your master's, produced a 28 gene chip for the American eel. And so this has not been printed or tested, but those assays are ready to go if partners do in fact need them. And then some of the work that's been going on at the University of Manitoba in my lab and with Margaret Docker, we've been producing a 56 gene chip for sea lamprey. This was designed by Alyssa Weinrock and then tested by Ellie Hung. And so this was looking at more general stress, thermal stress, and contaminant stress for sea lamprey. And contaminant stress is very relevant because of the chemical pesticides that are used in sea lamprey control. And so from this initial work, we were able to get 44 of these genes to amplify in the gill, 47 to amplify in the liver. And so then we took the ones that performed well and then added in some new ones. And then these are now being refined for a version 2 of the sea lamprey chip. And then we also tested out how well these assays that were designed for sea lamprey, how well they worked in northern brook lamprey, which has no available assembled reference transcriptome. And even then we were able to get some of the assays to amplify in this other species too, which is again quite exciting because as Margaret will tell you, lamprey genomes are rather complicated. And so now we have we're trying to test this out in a study which I'm quite excited about, which is where we've got sea lamprey that were either fasted or they're being fed on rainbow trout. And we've got also, so we'll be able to look at how their transcriptional profiles change using the chip based on whether they're eating or whether they're not. And then we're also, because we can, looking at the transcriptional profile in the rainbow trout that's being fed on by the sea lamprey. So it's going to be a kind of a fun, interesting, sorry, not fun, a very informative and interesting way of testing the sea lamprey chips. And then one of the things that came from all of this, as we've talked about yesterday, is whether we can start using tissues that can be sampled less invasive than sampling things like gill and liver and the brain for using the STP-chip technology. And so whether we can use things like mucus swabs, which Annalisa talked about yesterday, or measuring the RNA in the water from tanks. And one of the things that's important about trying to develop these types of approaches or try to use these types of approaches is that it's not always the right tissue for the question that you're interested in, especially something like a contaminant response where liver is going to be the best tissue, Daniel's favorite tissue. So what we've been doing a lot lately is just trying to compare what the mucus transcriptional profile looks like with other these more these lethally sampled tissues to see how well it represents that type of a response in the animals. And so one of the ways that we did this was looking at lake trout. We had lake trout that are acclimated to 4 different temperatures, and then we also had a group that was at regular oxygen levels and then also in hypoxic conditions, which are really relevant stressors for lake trout in the southern ends of their distributions. We used one of the salmonid hypoxia, thermal stress, and general stress STP chips for doing this type of work. And then we compared the gill, liver, and mucus from these fish. And then what we did is, well, this is work from Alyssa Weinrock, which will actually be published any day now in the Journal of Fish Biology. We then just try to do a sort of an overall correlation of how well the mucus profile related to other tissues, so whether related to the liver or the gill in both normoxia and hypoxia. And what we found is that the mucus profile using the assays on this chip tended to correlate quite nicely with the gill, which makes a lot of sense because, again, those are both tissues that are at the interface with the water, and then the gill has a tendency to show up in things like eRNA and everything, so it's shedding these types of transcripts into the surrounding environment. But this type of work suggests that mucus can be a useful tissue for identifying stressors in lake trout. And from this work, this, as well as the work that Annalisa talked about yesterday, was used to try to narrow down into a 28 gene mucus chip. So these are all assays that do in fact amplify in mucus swabs from these animals. And this is sort of going over again what Annalisa talked about yesterday. This was more of a focus study on trying to directly test the performance of the mucus compared to other tissues. The lake trout example was sort of an add-on, where this was much more with the specific goal of testing the mucus across the other tissues. And this was done on juvenile bull trout, and they're acclimated to 104 temperatures. And what Annalisa was able to show is that, again, the mucus profile tends to show, if you put it all into a principal component analysis, you can start to see it separate out from sort of what would be considered good temperatures for bull trout versus ones that start to become stressful. You can actually see that deviation in the transcriptional profile start to occur. And this is something that can be detected in mucus as well. And Annalisa also did, again, this correlation style analysis to see how well it represented transcriptional profiles at the individual tissues. And again, it tends to match well with what's going on in the gill. So again, quite promising that this is a potential tissue that will work. And then lastly, Shahin did some work out at Yellow Island Aquaculture. And this was using a 28 gene salmonid stress chip. And what he was doing is he was interested in trying to see if we can use water samples for transcriptional profiling. And so he had Chinook salmon that were held in fresh water and acutely exposed to either brackish or salt water, and then took gill, liver, a mucus swab, and then just a water sample. And of the 25 genes of interest that were on this particular chip, he found 6 of them could actually amplified in the water sample. So it really is a kind of a proof of concept that you can use this chip technology for eRNA, which is great. And it also shows that this could be a really useful approach for situations where you have fish in a tank or in captive conditions. So it's really quite an interesting and promising result. And so just to recap all that's happened in the last 5 years, we've produced a lot of these different salmonid chips. Again, we tried to design assays that sort of represented these types of functional categories, and then we could sort of modify them however needed based on the studies question that we were looking at. A lot of these cases, we've gone from a version 1 of the chip to version 2, sometimes version 3 of the chip. So it's modifying all the time. But again, it's actually quite a large number of these STP chips that have been produced already. And then this is just sort of a pretty way of looking at all that's been produced. And then just a reminder that it is an iterative process. So again, we have to design the chips, print them, test them, and then we improve them for the next round. So it's going to be an ever evolving thing. And then eventually someday, hopefully we'll end up with the final chip that will work well all the time. And I think that's all I have. So I'll pass it over to Daniel, who gets to talk about the application of these chips.
Yeah, so my job is to talk about activity 4. I always had envy of Ken because he only had one objective at activity 3. We had 3. And I want to remind you what the point was of activity 4 within GEN-FISH. Activity 3 was generating and validating the chips in the lab, mostly in silico. But the intention, of course, was to validate these chips so that end users would be confident in adopting them. So the second half, activity 4, was sort of the in vivo validation. And a bit of history here, when we put together the proposal for GEN-FISH, we got enormous pushback on our use of the word validation. We had a pushback when we submitted the proposal, and we had pushback when we had the face-to-face defense of our proposal. So the behind this is sort of the three objectives within activity 4, objective 4.1. This is lab-based challenges. So the idea behind this is we're controlling not only the stressor that we're applying, but also the environment. So this should be as clean as you can get in terms of detecting the response to the stressor. But that's not all that useful for our end users. So then the next step was field-based challenges. So these would be either in a mesocosm or a gradient of stressors. So this is where we know what the stressor is. We have a relative idea of the intensity of the stressor, but we're not controlling the environment. We have this background variation that might be messing up the transcriptional profile. And then this is perhaps where the rubber hit the road for activity 4. It was certainly something that the reviewers of the proposal liked, and that is the partner initiated projects. And the important part of this is it's not, we go to a partner and say, I really like liver and osmolar regulation, which is the bad combination, but that's what I want to do. So will you help me do this? It's a case of partners telling us what the problem is, and then we work with them to design the study, which may be more or less controlled depending upon the partner. And the idea behind this is proof of concept. So can a partner lead the project and still get information that's useful to them? So we're going to start out with objective 4.1. This is controlled stressor validation. It might be a single applied stressor or multiple stressors. And I've actually shown these at previous AGMs. So these are essentially just graphics showing the species on the y-axis and the stressor on the x-axis. What we originally proposed was 46 species and 9 stressors. If you do that calculation, as single stressors, it's a lot. If you then start looking at combinations and permutations of 2 stressors, it's an enormous amount. It was ridiculous. The other thing we discovered is a bunch of the species that we put on that list are of importance, but they're of conservation importance and there's or invasion, and there was no way we could get live organisms and do bad things to them and harvest their brains and livers and gills and stuff. So the number of species that we worked on was shortened, but it still shows diversity. My worry when we started this, it was all going to be salmonids, but we actually included a bunch of other cool species. As I explained to the ROC, this hasn't changed in the last year, really. Maybe 1 or 2 have been added. What's changed is it's gone from we have tissue samples in the -80 to we actually have STP-chip data for a lot of these. Multiple stressors is more limited. Multiple stressors logistically is a lot more complicated, but still an impressive accomplishment. Then we move into the field or controlled trials. This, of course, was really impacted by the delays associated with COVID. For a long time, this schematic was reflecting plans. We talked to people, we had plans, we had a design. We didn't even actually have samples. At this point, a lot of these projects have progressed to at least cDNA. In a lot of cases, we actually have the transcriptional profile data. And this is the final objective, 4.3. This is proof of concept. This one blew up on us, which surprised me. We originally proposed, I think, 8 or 9 proof of concept case studies, and we ended up with 16. And the reason being is as our partners became more aware of the promise of the STP-chip, they bought into it and came to us with things like the effect of angling on largemouth bass. That's something Steve Cook students or group followed up on. A lot of these have progressed to the STP-chip. Some of them have projects are completed. And this final on the far right there, the status of the project, that I suspect that's out of date. There are probably a lot more papers, reports, or theses completed on that. So that's the overview. Now I'm going to give some examples. You're going to have already heard about some of these examples because what I did is I cherry-picked ones that provided an evidence of diversity or interesting applications of STP chips. So some of these you've already heard about in detail yesterday, or in summary with what Ken was talking about, or even over dinner last night. So the idea behind I'm not giving you details on this. If you're interested in details, we can follow up on them. So first of all, the non-lethal sampling. This was not in our proposal. We were planning on killing fish for activities 3 and 4. We started getting pushed back to do non-lethal sampling. So gill biopsy, muscle biopsy. There was preliminary work done out of Nicolas Bernier's lab and another lab. I can't remember where. Could we take a gill biopsy or muscle biopsy and not kill the fish? Which we can. We sort of knew that, but we validated that. We went further than that. They were interested in potentially non-invasive, which includes like a mucus swab, either on the side of the fish or the gill swab, and environmental RNA. I'm not going to talk about any of these. Well, I actually am, 1 or 2 of them, but these are the projects, applications of this non-invasive that I know of. So the first one is work by master student Holly Moscow, working with Tina Semyuk in Atlantic Salmon, Nova Scotia. She's applying the swab mucus chip to the Atlantic salmon. Shahin Islam, you've heard about Shahin already. He collaborated with BC Freshwater Fisheries Society, and they transport 3 different types of salmonids to lakes and release them. They were interested, could we actually identify stress levels associated with that transport? And in that, he did the swab and environmental RNA. Thermal stress in wild bull trout. This was analysis stuff, where it's an actual application, not just validation. And then Shahin also did work with Yellow Island aquaculture using eRNA and swab. Ken talked about the eRNA and what was it, 7 genes that worked? 6 genes. Shahin decided that wasn't very good. He wasn't very happy with that, even though it did manage to identify the different salinity challenges. He's really excited about the transport stress. These fish were much, much more densely packed in the tanks, and they were they'd been in those tanks for months as they were reared. And he's saying that the environmental RNA from those rearing tanks at the Freshwater Fisheries Society seems to be working a lot better, but he hasn't analyzed that completely. So I'm just explaining that this is, we not only develop the technology to be able to consider non-invasive, we developed the technique in a chip in Ken's lab, and now we've got 4 solid applications that are in various stages of analysis, from being complete to Holly's out there wrestling these fish as we speak. Okay, so examples of single stressor trials in the lab. I'm going to talk about my student, Nick, who worked with Yellow Island. I'm going to talk about Carol's work with the bull trout. So Nick Tugi was a master student who was at Yellow Island Aquaculture, our only commercial salmon, commercial fish culture partner. He used the osmoregulatory stress chip. I'm not going into detail. It was a complex design, but one of the things that Yellow Island was really interested in is what happens if you take 2-year-old Chinook salmon from the salt water and dump them into fresh water. The reason being is that DFO had shut down their saltwater cage facilities, and they had to move everything into their freshwater rearing facilities immediately. And so they wanted to know whether that was affecting them. They didn't die, but was it affecting them in terms of their fitness for their ability to survive and thrive? So what you see here is a list of the genes on the chip, and the various, the y-axis gives you the effect of treatment, time, and time by treatment, because he sampled the fish over time. And a couple of things that I'd like to point out. So if there's gray, it means it wasn't significant. If it's colored, a different level of significance. First of all, there was definitely a treatment effect on these guys. They noticed. The other thing that was very interesting to John and Ann at Yellow Island was that 3 of the 4 reporter immune genes were showing a response. They didn't expect this, but it was what they were worried about, that the transfer didn't affect them immediately, but was triggering some kind of immune challenge. Okay, and I'm going to talk about Carol's work with the bull trout. And again, the bull trout are a species at risk across a lot of their distribution in Canada, the west of Canada. And a big problem for them is they're a cold water species. We've heard about that yesterday. And the question was, how are they going to respond to temperatures above their preference? And so the experiment was taking bull trout and acclimating them to 6 different temperatures for 3 weeks, and then essentially assessing their transcriptional profile. Ken, you might be a little horrified by this, but here is all that Carol did. The paper that's in press at this point, this is the only thing I took from it. And this is sort of a PCA based on the transcriptional profile. The 6 temperature treatments are shown there. You can go into details and dive into which specific genes are driving this variation. But what's exciting to me when I saw this is you can tell the different, not only is there temperature stress, but you can tell the difference between a 21-degree temperature stress and a 15-degree temperature stress. Very encouraging for using transcriptional profiling. Okay, controlled multi-stress trials. So this is Kyle with Nick Bernier and Ken's lab looking at food deprivation and thermal stress. So 2 simultaneous stressors with lake sturgeon. So this is Kyle's schematic for his project. Needless to say, I'm not going to go into the details. What they did is they fed the sturgeon and they starved the sturgeon for 5 or 10 weeks. So this makes it more and more complex. Clearly, they want them to do a lot of work. And then they either collected tissue from them immediately, or they challenged them with the CT max, so thermally stressed them. So the idea behind this is, first of all, is the temperature stress going to cause an effect? And is there going to be an interaction between fasting and temperature stress? And again, apologies to God. There we go, his whole project with 1 gene, lactate dehydrogenase. The point that I'm trying to make is we have the fed sturgeon, the fasted sturgeon, fold change, and then the red is the control, so they didn't get temperature challenged. 2-hour and 24-hour temperature challenge. This is hours after the CT max. And the point that I want to make from this, that again, I thought was really impressive, is that the feeding history affected the response of the fish to the thermal challenge, and quite dramatically. So the fasted fish said, man, don't care about the temperature challenge. There's virtually no transcriptional response to this 1 gene, but the fed still showed this robust. I was just talking to Nick about this, and there's a lot more behind this, but it highlights the fact that there's an unexpected interaction between the 2 stressors. Okay, now we move into the field or mesocosm, where we can't control all of the environmental parameters, but we can either know about applied stressor that's occurring naturally, or we can do it on purpose. In both of these cases, there are gradients. So Patricia, we saw her 5-minute talk last night after dinner. She used her own personal, private, favorite osmoregulatory stress kit for perciforms that involve temperature, immune, detoxification, hypoxia, metabolism. First of all, what she did, you know about this, Patricia, right? Correct me if I'm wrong, if I make a mistake. She did a controlled experiment with farmed yellow perch. And so the baseline here was about 19 degrees, I believe. And then she exposed the fish to other, replicates to a 22, 25, 27 degree challenge. And then looked at the transcriptional profile across all of these genes. Okay, so what you see here, first of all, which is again very promising, very good for Patricia's species, but for me at a bigger level for activity 4, is you get this characteristic transcriptional profile. So this is up and down regulation relative to the non-temperature challenge, the 19.5 degree ambient temperature. And you can see that there's a characteristic switch, and the pattern, if you squint your eyes, is discernible. So you get strong changes at 25 relative to 22, and then 27 seems to be going back down or back to yellow if you squint your eyes. And all the stars show the ones that showed a significant treatment effect. And when Patricia zoomed in on one particular gene, glucocorticoid receptor, so general stress, she found this interesting pattern, whereas you went from 19 to 20 to 22 degrees, there's an increase in the expression of this receptor messenger RNA. When you got up to the 25 and 27, you start seeing downregulation. This is consistent with early work that she'd done in the field where she saw the same thing, an adaptive response and possibly basically transcriptional breakdown associated with approaching the lethal limit for these fish. And then she described yesterday her spatial gradient project in collaboration with OMNRF. And she described this better than I can, where she identified these two lakes in northwestern Ontario that were showing a very divergent transcriptional profile. Remember, these are just captured fish. These are not challenged. So this is basically transcriptional profile in the lake. And the reason why this really interested me beyond what Patricia described is this, to me, highlights the beauty of transcriptional profile. There is absolutely no reason why you'd identify those two lakes as being potentially unusual environments for yellow perch. But the transcriptional profilers are saying, hey, look at me, something's going on. And as Ken and Nick and I were discussing over breakfast, we haven't got a clue what it is. We might be able to interpret it based on which genes are up and down regulated at the transcriptional level. But essentially, it's telling us, look at me, which I frankly did not expect. There's something going on there, probably not temperature related, but it might be. Okay, this is Holly's project. As I said, Holly is currently in Nova Scotia doing her field work. And the Atlantic salmon in Nova Scotia are in trouble. There's a lot of concern about stocks. And one of the big issues they're facing is warming temperatures. The Atlantic salmon don't like warm temperatures. So we've got the climate change issues, but you also have introduced predator, smallmouth bass. So you've got this dual or two contributing stressors, and they occur across a gradient. So within the study area, it goes from high temperature and high levels of smallmouth bass predation, we assume, because there's lots of smallmouth bass there. As you go more upstream, the temperatures go down and there are fewer predators. And so what Tina and Holly have come up with is they're going to swab these fish and use the mucus salmoniid stress chip to see what the synergy effects are of temperature and predation pressure. And then the part of Holly's project that I think is really cool, and this is the only one in all GEN-FISH that does this, is she's also using eDNA. And she's going to do eDNA primarily because I said, why don't you use it to determine how many smallmouth bass are there? Because it's one thing to say high smallmouth bass, low smallmouth bass. What if you use eDNA and you get an actual copy number? How cool would that be? And then Holly came up with the idea there's also a possibility that there's an alternative prey that occurs within the system that with there at high levels, then perhaps that would reduce predation pressure. So the same eDNA can be used to quantify the alternative prey. And off the hand, I've forgotten what it is.
Alewife.
Alewife. I knew that. Okay, so I'm going to give you 3 partner-initiated projects. And I try to do them quickly here. So first one, the partner is DFO West Coast, and the GEN-FISH person is Renee Kuplicate, who is an undergraduate thesis student, a contemporary of Kyle's. And the project was initiated by collaborator Kyle Wellban, not to be confused with Banwell. Main reason I recruited you, Kyle. No, so Kyle Wellban is a former PhD student of mine, and he's now on the West Coast, and he is concerned about climate change impacts on Chinook salmon, among other species. But one of the things he's always been interested in, as have I, is the process whereby DFO Salmon Enhancement Program take fish from one river and dump them into another river to supplement those populations for harvests. If you move them, they're going to hybridize. The natural belief, or the widely held belief, is that all of these stocks of Chinook salmon are locally adapted to more or lesser extent, and that when you hybridize them, you're going to get a hybrid that will have lower fitness within the single environment because they don't have the proper co-adapted gene complexes. So what Renell did was he used the 56 Salmonid general stress chip, and he used it on all possible combinations of hybrids. Go back to this. He had 4 populations that DFO were willing to provide gametes. DFO West Coast crossed them in all possible process. So you had pure knit-net and then hybrid knit-net with all 3 others and just all other combinations. And so what he was interested in is 2 things. First of all, do these fish in general respond to a thermal challenge, transcriptional? And secondly, does whether they're a hybrid or non-hybrid, pure type, does that affect how they respond to the thermal challenge? So this is only 6 of his 52 genes. But what's interesting, so there's a couple of examples of temperature stress, metabolism, and immune function related genes. One of the things that was interesting is this very consistent pattern of response to the challenge. So on the left, we have a control that's no thermal challenge. On the right, we have a 2-degree thermal challenge for 1 hour. This is at the just at the swim-up stage in the incubation of the salmon. This one's consistent across almost all genes. In some cases, all of these are upregulation under thermal challenge, some cases downregulation, but consistently these fish were definitely responding to the thermal challenge. The interesting thing is, consistently, the fish didn't care whether they were hybrid or pure type. There was no significant difference in the transcription of the genes depending upon their genetic background. This feeds back into DFO's concerns. The concern should be the temperature challenge, not the genetic background of the fish. The next case study I want to talk about is work done in Mark Shrimpton's lab by Carolyn. Again, she gave the presentation yesterday. But this is factors leading to the decline of sockeye salmon in the lake in the Gittin territory. And so, as Carolyn described yesterday, essentially the way that the approach they took was the Babine Lake sockeye stock is in good shape, the Gittin is decline. So let's compare the early stage fish to see if they have different transcriptional profiles. Carolyn used a couple of different chips. The one I'm going to describe the results from was the osmoregulatory stress, the OSTP chip. And Carolyn showed the same graph yesterday. And this is where I'm departing from Carolyn and Mark. Because Carolyn said, basically, these fish are fine. They're the same as the baleen. So if you look at this, the blue dots are the baleen sockeye juveniles, which are doing fine. And the green ones are the juvenile sockeye, and they're not doing so well. And when you look at individual genes, none of them are jumping out as being differentially transcribed between these two groups. But two things to look at. First of all, PC1 versus PC2. No significant difference for either principal component, but what jumps out at you is the variation. So what my interpretation would be, why the heck are the GEN-FISH showing such controlled transcription? This is classic response to stress, where you start clamping down on the scope of response. So to me, that indicates something may be going on at a whole genome level. And then when we look at this, we have one PC that was significantly different. Was it 3 or 4?
It's number 4.
It's significantly different. But even if you don't care about the genes, and I know physiologists have a hard time doing that, but if you say, I don't care what the genes are, you see this shit. This is close to diagnostic. You might be able to do stock ID between baleen and GEN based on that PC plot. Something's going on. Is it osmoregulatory stress? You rule that out by doing other assays. But there's something going on there. There's something fundamentally different in the biology between these two groups, and it highlights that maybe more targeted gene expression should be done. Okay, the last one I want to talk about is Dane Roberts' project in Trevor Pitcher's lab. This is the person that Trevor reported just got a job with Ontario OMNR, so good for Dane. He did a great job. And what his project was about was very similar to what Shaheen was doing with the BC Freshwater Fisheries Society. They're growing Atlantic salmon, they're loading them in transport trucks, they're taking them and dumping them into Lake Ontario tributaries to try to make the Atlantic salmon come back. And what Dane did was he sampled the fish in the tank before they even touched them. After they loaded them, so they had the loading stress, and then after they've been transported and were just getting ready to be dumped to their target. He used the general stress chip, 28 gene, to assess transcriptional profile, and only 2 genes came out as significant. Significant between baseline, loading, and transport. Both of them are related to stress, but you can see that for ENO1A, there was a significant increase to the loading, but then they recovered during the transport, which is pretty surprising to me because the transport is stressful. But IGF-2 did what you might more readily expect, is that there's cumulative stress response in terms of the transcription of this particular gene. When he looked at cortisol, which is the classic gold standard for stress in salmonids, got exactly what you'd expect, cumulative response in cortisol levels in the blood of the fish. But what this indicates to me is there's more going on in simple stress. Lactate levels that Dane also measured tended to follow the pattern of ENO1A. I think that a more in-depth exploration of the actual transcriptional pattern might have shown a different pattern along the lines of what Carolyn did, where you try to interpret the pattern of transcriptional profile rather than gene by gene, because these are the only 2 that came up as significant after Bonferroni correction, which I disapprove of. Anyways, And it highlights the need to move on from what we did in activity 4 into actual patterns of transcription, recognition, and interpretation. Okay, so the outcomes or the questions of activity 3 and 4 is, first of all, sampling, which is the first step of transcriptional profiling. You can do traditional, and it's really valuable in some situations where you can sacrifice fish. Non-lethal is another option. It tends to be the worst of both worlds. The people see it as you're poaching holes in my fish. I don't like that. And Tina and Holly have run into this with the Atlantic salmon par. And you're still not getting the ideal sample. You can't get a non-lethal brain sample or heart or liver sample. But then there's the non-invasive. Now you can choose traditional or non-invasive, where you just do a swab. You're going to lose information if you use non-invasive, but you've got other benefits that counterbalance that cost. Application. 2 concerns: cost and facilities. Availability of STP-chip analysis. These are the cost. I don't think is a big deal. I think that if the salmonid general stress chip that activity 3 developed, 56 gene, becomes widely used, then Shelby can order in like 1 million of them and just chug through them, and the costs will come down. The facilities are going to be limiting. If everybody decides to adopt that, you're going to be busy, Shelby. And then interpretation. The sort of go-to direction is single gene. And Nick Tookie just defended. And his first version of his transcriptional profile chapter is, this gene did this, and then this gene did that, and then this gene, and you go, oh my God, this is going on forever. Can't imagine the 56 gene chip. He only had 28. So we have to start looking at the gene interactions. That's it. Oh. So we both stand up. Tina.
So in terms of the applicability, I'm really thinking of the profile is going to be really important to go after. I could just think of that starvation of a sturgeon example. Had you just looked at that profile in isolation of a challenge, was it that, you didn't know anything else? You said, oh, I think they're being affected by temperature. And like, well, they're not responding at all. These fish are okay. But without having, let's say, your appetite genes to show it's also fasted, you could have made an erroneous conclusion. And so were there backup genes to show evidence of fasting as well? Absolutely. Yeah. So this is why I think pushing multiple genes at the same time, but therein potentially lies the issue in how you interpret it, because it's nothing if not messy as well. So do you have any advice going forward? This might be more for the afternoon, but given the importance that when we're working with end users who may not know the stressor, or they think they know a sole stressor, when confronted with the entire profile, how do we help them interpret that?
So I'm going to give you the bad answer, and then Ken and Nicola can give you the right answer. My view, when we're talking about this, is the transcriptional profile lets you know something's going on. The next stage would be, how do you interpret it to say what is going on? It's a huge step forward to be able to say something's going on. And that's why, the preliminary, those results that I showed from Kyle were worrying to me, because exactly what you say, the combination of the fasting and the temperature challenge, you would interpret it as there's nothing going on. Your immediate response wouldn't be there's two things going on, because they cancel each other. They don't, but you know what I mean? So that's where the profiling comes in as being important. But in terms of actually interpreting the profile, Nicola and Ken, in terms of Kyle's work?
Yeah, the whole profile is incredibly interesting. And that's, we did some physiological assays too, which gave us insights and interpret the gene expression profiling. But the whole data set pointed in some directions and like it pointed in the direction that, for example, the fasted fish would clearly had more cellular damage and more oxidative stress damage because they had shut down a whole bunch of defense mechanisms because of this long-term fasting. But I would be so reticent from drawing absolute conclusions or not being able to, do follow-up studies and show, yes, there were clear cellular damage and so on and so forth, just based on the profiling. But the profiling, just like Daniel said, suggested that there was a problem. So if nothing else, you can identify the problems. Next challenge, I guess, is to then determine the magnitude. Do you have a problem that you need to do something about? That's really kind of the next.
That's more complicated than one.
Yeah, I'm like, Annalise said something to say.
Not a lab study. I was thinking about my field stuff, so like if you see groupings, like you measure the wrong thing in the field. Like I look at thermal stress, but then recently someone sent me a map of all these oil spills out on my site, so like, oh my, I have so many variables to put into my models. And then if nothing significant, but you have these groupings, you can say something's wrong, but I don't know what is driving it.
It's similar to sort of my alternative interpretation of Carolyn's PC plots, where there's something different.
Yes, I was going to ask you about that. Because the Bemine has a population of about 1 million potentially salmon return to that. There's probably 15 to 20 different distinct populations with Bemine.
Mm-hmm.
That's one spot on the game. So you just said that from a much more conventional. Do you buy that or?
I was gonna say the same thing. Just like plasticity or diversity.
Most of the genes on the chip are not particularly affected by taxonomic divergence. They, could you use the chip to generate a phylogenetic tree? I bet you could. I bet it would be really cool too. But my point is I don't think it's the stock structure, but fundamentally you're looking at N of 1 and N of 1. So I don't know why that higher variation is there, but it does jump out at you as being, is it the fish that are showing divergence of transcriptional response? That is, they're all going in different directions? Or is it that they're all being sampled, they're inhabiting different habitats?
Are they a starfish that get you?
No, they're all natural spawns.
I think it's what reveals them too. Right, that was a much broader geographic scale, and it wasn't stress under control as well, but like an even much larger geographic scale population had that variable with the transfer from both populations. Experiments.
I didn't show that, but he looked at sort of the pure response across the 4 populations, and they were all, their profiles were all identical.
Yeah, I mean, it's super complicated, and I think just because I've been hearing a lot of the same criticisms of transcriptional profiling. No, I'm not. I mean, this other person.
Her name starts with Chris and ends with
No, it just, but people have been, kind of saying how, well, you can't really hang your hat on transcriptional data and all these things. And so we've always, at least for lab studies, we're always trying to add in those other sort of well-known markers so that when you have a transcriptional profile, if a bunch of anaerobic metabolism genes are being activated, and you also have the plasma lactate to sort of support that, okay, it looks like that's all collectively showing a pattern that's all pointing in one direction, as Nick was saying. That's sort of how we try to at least build a weight of evidence approach, of saying that, you know, something's happening, it may very well be this. But I think, you know, when it comes to the field studies, because there's just so many possible variables, I think, you know, it's a great way of, as Daniel said, you know, identifying spots or populations that are suggesting we need to take a deeper look into what's going on with those fish. And screaming out the, hey, look at me kind of signal on those is sort of the what I think would be, you know, would be a great application of this approach. And then I think the huge advantage of these chips is that we can run much larger sample sizes, much faster, much cheaper, so we can get a very quick overview of what's going on, and hopefully then from there we can identify where to focus our efforts.
Well,
So really kind of unrelated question. So in terms of interpreting what these transcriptomic patterns are telling you, again, you showed two examples. Should work with thermal stressors and purge. And you sort of started here and you kind of fell off, and you interpreted that as these animals were basically falling apart transcriptomically. They can't respond anymore. Then like Dane's work with transport stress, and profiles went up, came back down, you interpreted that as, oh, they're recovering. I mean, a classic kind of like response in toxicology is hormesis, where you get a response to the animals, okay, I'm going to take this challenge on. Then you get past the point where they just can't do this anymore, or they're capacity advantage and close things down. So I mean, it comes back to, I think, something that Nick mentioned, Tina's question, sort of how do we interpret these profiles when it's like, okay, we get to this point, this is animal collapsing, and we think that because thermal stress, but we know where the thermal stressors are. How do we end up interpreting whether something is up and comes down, for instance, as fluid or vacuum? Does that come down to measuring the GEN-FISH 2.0 performance and fitness and things like that?
well, comparing Dane's and Patricia's patterns, Dane, the fish were in the same environment over time. So it wasn't that you were getting sort of different challenges, I guess is what I'm trying to say. The interpretation of potential for the first gene, where essentially it's just lost its scope for response. So stress is still going up, but it's simply plateaued. Sort of like happens with cortisol. It plateaus because it can't do anything more. It's hard to know. And it's Dane and Trevor's interpretation of that was that it was basically plateauing because they were acclimating.
I mean, knowing a lot about your gene tells a lot. You know, what is the capacity and what is supposed to be doing? That gets you a lot of them, like, it's really, you know, what's my criticism at all here? Just curious, you get these patterns as a total noob, like me, you know, if I were to send samples off and not be go back, I would need a lot of help, not being an expert, to interpret the patterns, what it means, you know, and I probably would need help from people understanding the performance of the organism too, to know what this thing is doing. You know, as the genes are changing at time. Sorry.
No, I just got excited because you brought up toxicology, and that's where a lot of the sort of framework that we use in my lab come from, because they did a lot of ecotox work as well. And so that the whole non-linear response patterns that you see is super exciting, which is why we sort of almost to death, we do those acclimation studies over, you know, different temperatures that span the whole thermal distribution. So we can try to identify where you start to see the ones that kind of go up and go down, and then versus ones that start to, you know, do the really dramatic okay, this is really a problem and we're starting to, it's really going to be an issue. So I try not to swear with how I describe it usually, but I call it the oh shit response. And that's where like, things have changed from an adaptive response to a much more extreme. This is a really severe thing. And that's why I get so excited about what Carol had produced, where you have, you can tell the difference between a 21-degree stress profile and you have a difference between, 15-degree stress profile. And I think that's super exciting. And then some of the new analysis approaches that Annalisa's brought in, where you can now start to see where you get a break point and where that starts to change. So after me just kind of waving my hands for a while, she actually has a way to quantify that this is actually happening, which is really exciting.
So you didn't like my idea of just squinting at the pattern? You want to do it online? Or are you cutting us off? Yeah. Sorry, Nick, got to move on to the most important activity.
Thank you, guys.
I was gonna try to look slowly. Okay, I was gonna try to just swivel this a little bit. It's okay. And swivel this trench. Sorry.
Because I might not stand in front. No answer for Daniel. He's going pink. I can start very slowly. So activities 1 and 2 and 3 and 4, like we'd mentioned yesterday, they are meant to produce deliverables that were applicable to our co-producers. And so this is what's reflected in our hats that Joe pointed out. We've got a fish survey toolkit. We have a fish health toolkit, and our toolkit is all about a decision-making toolkit. And this is what we pitched to Genome Canada. This is what they liked about it, because it really fell into a section that all Genome Canada grants have, which is the GEL section. So this basically stands for the genomics and the ethical, economic, environmental, and legal applications. We mentioned yesterday that we dropped the L because it wasn't actually really applicable, which leaves us with the G's kind of acronym going forward. But before we started, I know that a lot of us, we have a lot of partners. We have a huge network. And when Daniel was saying that activity 4.3 really took off, it took off before Genome Canada actually started. This was the impetus for our Genome Canada and GEN-FISH grant, is that we had already our co-producers from the beginning, which is really interesting because the language that Genome Canada uses is end users, which means that there's no involvement in anyone. That's what the implication is, which is completely antithesis to what GEN-FISH is all about. We already have, again, like I mentioned, our partners who are uptaking these. They're the ones who kind of said from the get-go, this is what we need, this is what we want to look into. But these still can be a select few. This is not a broad categorization of all the sectors that we're working in and all the co-producers, again, that we're looking to recruit. Daniel even mentioned we only had one commercial fisheries, which was Yile. We didn't really have as many, let's say, fish cultures throughout all of Canada. This actually is why we bring in this GEL section. It's social science, but really it has to be interwoven with the rest of the activities to truly be effective. So that's why the very, very end product are these decision toolkits, which are still kind of somewhat in their infancy, I'd like to say at this point. But I'll tell you more on how we've gotten to this point regardless. And it was to basically assess the utility of our general toolkits, identify barriers to potential adoption. Now, I might be thinking that's great. We're just going to survey fisheries managers. We're going to survey NGOs. We're going to survey environmental consultants. But the fact is we need to start with general public. And this was taken under the umbrella and the wheelhouse of Dr. Amy Fitzgerald, who is at the University of Windsor, along with her PhD student, Jennifer Halliday, looking at emerging ethical concerns. And she really did, for her first part, target the public, because again, sometimes our fisheries managers are dealing with and have to have their approaches acceptable by the public, or the public dictates what they like. Whether or not you're working with NGOs are always looking for funding. Funding comes from the public. So what I'm going to just give you an overall findings. I have presented this data before in previous AGMs, but I'm happy to summarize it all again.
So the first thing that Amy did, she did something called a media sweep. What's out there in the media, both in the media that we find in Canada and the United States, because oftentimes media can dictate what the public should be thinking about, or sometimes the media echoes what's going on in the public and tries to present it more globally. So she was looking for any kind of ways and themes in which eDNA technology was situated and interpreted and basically written about towards the public. And then she did a second one looking at transcriptomics, and not necessarily genomics, proteomics, metabolomics, transcriptomics in terms of gene expression, how that was positioned in the media. Her two main findings were very much like the public is not really aware of it. In terms of the eDNA, the only thing that really stood out in one of the articles over the past 20 years in the media was actually related to fish, and it was using these eDNA methods to identify invasive Asian carps and how it was used in a case to show that they were present and it would enact some form of management and retention in terms of the United States. When it came to transcriptomic messaging, it was only ever related to what she referred to as the molecularization of life, which meant it came down to GMOs, genetically modified organisms. So the public really knows about it in terms of gene expression and how to interpret it, in terms of how it's going to almost be like a Frankenstein type of monster and how it could potentially change genomics. That was the only ethical issue that was raised. Again, not very present. And you're going to see this as a common theme. This is a common theme throughout the entire GEN-FISH in terms of stacking eDNA against transcriptomics. From there, then Amy started to focus in with focus groups, fisheries managers, different sectors. She worked with the Chippewas of Nawash First Nations group, their own focus groups. And there were some themes that were unique to different sectors and some that were common. And some of them were, we talked about this informed consent about collecting environmental DNA through water samples containing human DNA. But there was also the concern sometimes with certain sectors. And again, her report isn't public yet, so I don't feel comfortable speaking about it until it's been approved by all parties. But one of them was using this information from eDNA to have commercial interests supersede the interests of conservation. So in the sense of, oh, no, this is really important. This is a commercially important fish. It's found here. This is much important to do rather than, let's say, habitat restoration. Just to justify, using these tools as a way to justify interests of those who are more powerful amongst the different sectors involved in fisheries, conservation, and management. Mostly, again, focused around eDNA, which is a little bit more diverse. When it came to transcriptomics, this is where that kind of theme of manipulating what an individual means emerged again amongst the sectors themselves. And it turned into whether or not we're going to be selecting fish that show, let's say, from a different population altogether, that potentially show a greater resiliency or resistance to environmental stressors, bring them into another population and have them take over. So it's a way of potentially indirectly manipulating what it means for animal welfare. So that was a concern that was, again, specific to the transcriptomic side of things. So that was Amy's work. And she's been invited to the Chippewas of Nawash salmon ceremony. Her results have been accepted by the environmental coordinator group that agreed to do it. And she's just making sure that now the community is fine with the way she's not just captured the information, but how everyone's collectively interpreting it. So that was Amy. The next is our economic side of things. And this was led by Dr. Tongji Lee from the University of Guelph. And here were her students that worked with Daniel Boyd, Connor Smith, Ian Boyle. And for one of her studies, Tongji looked at targeting recreational anglers themselves. And again, it was prefaced around eDNA. And she asked the question, how much are you willing to pay a premium to fish in a water body where it's being actively monitored using technology such as eDNA? And overwhelmingly, the recreational anglers were, this is great. I would actually pay anywhere between a 7% and a 33% increase in a premium to know that it's being actively monitored using this technology, which is really positive. The recreational anglers themselves felt very confident in their knowledge about what eDNA is and how it's used. So there was basically no uptake with recreational anglers whatsoever. Another aspect of Tongji's work was to figure out, okay, well, what makes an individual group or person Be an early adopter to a novel technology in which not much is known or is applied. What are some of the strategies? Because I mentioned yesterday that Tongji works in economic behavioral nudges, and sometimes these are like incentives, subsidies to increase uptake. And we all know that some of these technologies, they start out really expensive. So Daniel, 10 years ago, how much would it cost to run a single sample for a single gene for transcriptomics? And now?
On the open array, 60 cents.
60 cents. So $5, Daniel said, versus 60 cents. So there's two strategies individuals can adopt. One is to just be, I'm buying in really early. I'm an early adopter because I understand the worth of it. And then others will see an early adopter and then adopt it themselves. So there's a nice chain reaction. The second strategy is just wait. Wait until the cost comes down. Wait until maybe Thermo Fisher is going to offer a 50% discount on any of the samples that come through then. So it's a waiting game. But the worst thing that one can do, Tongji found out, is to play both strategies. Don't offer both. So if you're trying to promote early adopter, this is cool, this is amazing, this is really interesting, then don't also let it be known that there could be subsidies coming, because then everybody's going to wait without that knowledge that is actually going to happen. So be distinct. So again, care must be taken in incentivizing producers in those scenarios, which we thought was really, really neat. So information to go back in terms of commercialization and how we also, amongst our networks, how we position these tools that we're trying to get adopted. Before I get to what Dr. John Lieberman did, I also wanted to talk about the COVID pause that we all experienced, especially with our Activity 5 members who had to wait for a lot of this information in terms of talking through the development. We actually saw yesterday how much over the past 4 or 5 years the technology has evolved, the acceptance has evolved. So it really was we had to wait to see how we can get access to net partners, especially when the relationships were also put on pause during COVID. Well, we didn't rest on our laurels. We did something that I like to group in a theme, such as co-production of emerging technology. So these were a lot of multiple PIs, end users, as well as other colleagues across multiple sectors where we were exploring these questions. And I just want to do a shout out to Dr. Stephen Cook, as well as this recently graduating independent PhD student, along with Vivian Nguyen from Carleton University, Dr. Andrew Hallworth, who was really involved in a lot of these papers that came out during this time. And Steve mentioned it yesterday, these are thought pieces, but the take-home message here I just wanted to say is we actively work with our co-producers, Genome Canada's end users, to explore these themes, and again, co-production themes, emerging technology themes, and their intersection. The first thing that I think the earliest thing that came out from GEN-FISH was this paper that we put together. It came out in 2022, and it was looking at it from the transcriptomics, so activity 3 and 4. We work with our end users that span Indigenous community, which also span environmental consulting, with all of those span our commercial fishing. So that was Yael, John, and Anne. And we looked at what were some of the reasons why people would adopt this. You know, based on what they do, how did they think that transcriptomics was going to be important to their work? And this is when we came up with this nice diagram of what it looked like. So this is an early diagram, so you can see we're sampling muscle. So we actually have a tissue known as mucus or blood. And we're going to produce this lovely little heat map. We can tell you, oh, look, it's hypoxic, they're under stress, starvation, or their immune response. So again, it was like, it's going to be a nice clear cut. But we really realized that we needed that kind of figure so that our end users, co-producers, understood what transcriptomics is. And on the other side of things for activities 1 and 2, Nick already mentioned this paper that was led by his postdoc, Dr. Kenny Sperno, as well as other members of GEN-FISH and PIs at HQP, as well as our co-producers, looking at environmental DNA applications and challenges. When this came out, and again, I was just reviewing it, and one of the challenges that was expressed was Canada is so biogeographically diverse, it's going to be tough. And Matthew just showed us what we got there. Absolutely not. And however, the paper also mentioned that, you know, Canada's pretty wide, it's really far from communities, it might be hard to access. Proved that wrong again, but also proved it right, because for some reason we didn't get anything from PEI. Like, that makes no sense. So, you know, like taking that little crystal ball and looking into the future, Those are really good. And let's not forget that this is something that also contributes towards looking at the adoption and the acceptance of these tools. So whether or not it was exactly part of our mandate, this is something we really, really capitalized on. Again, ROC always nailed us about where are your co-producers? And we're like, well, they're here. We are working with them. We might not be working with them on, gene IGF/1, but we're working on the concept of adoption of these technologies. So here we've got just two papers expressly looking at co-production and fisheries and co-management. We've got this other paper led by Steve talking about ways to enhance the implementation of knowledge, let's say that's kind of held by these academic institutions or governments, and then the applicability of it, and then take-home messages, co-produce from the beginning. We have to do that straight out of the gate, and this is exactly what GEN-FISH has always underlines. Here's some more where we talked about technical science, and Dr. John Lieberman was on this paper, because it was mentioned in eDNA and the cost of adoption. How do we manage Canadian freshwater fisheries, challenges and emerging opportunities, revisiting the first report. Again, it all comes down to this is really good technology, but in order to do it effectively, we have to do it from the beginning. But not only that, we're working with so many different communities and sectors. We always have to ensure that we're doing, again, technoscience, novelties. It has to be meaningful and it has to be well-intentioned. So when we're working with communities, to always keep that. Are we addressing community concerns? Are we doing so in a respectful manner? Are we doing things that try to reduce harm as much as possible? And you've seen this today from activities 1 through 4. This is exactly what we've been trying to do and capitalize on. We have been listening to the needs from the very, very beginning of our co-producers. But then again, with Andrew's work, again, you were like, oh, that's very, very broad. It's like a meta level. But Andrew really did try to tunnel in terms of the fish survey toolkit and the health toolkit. Again, with all these supporting cast members, not the ones that we usually look to be the early adopters, like the government and some, whether or not it's the Fed level or the provincial level. But who are the other ones? Indigenous groups, NGOs, academics, environmental consultants. It's a no-brainer for the fish survey toolkit. It's very, very strong acceptance. Especially to help with the monitoring. And again, I think a take-home message I learned from yesterday, it's not like eDNA survey techniques are going to supersede anything, but a lot of the data coming out saying we should have multi-methods to still help fill in some of these gaps. When it comes to the fish health toolkit, it is going to be more difficult to adopt. Again, we need specialized teams, interested parties, ones that have like not just fundamental research questions to explore, but also have the capacity and the facilities, like Daniel mentioned, to be able to do that. So there's still going to be a challenge. So this all leads us to then, how do our co-producers make their decision in terms of whether or not they should be incorporating eDNA as a technology with their monitoring for a survey, or whether or not they should be adopting transcriptomics. So this is where Dr. John Lieberman comes in, working at the University of Guelph. Here, Mark, who is his research associate, who has a physics degree, working on one of the 2 toolkits. And the first one was this decision-making one. It was tested with end users, specifically it was fisheries managers. We now have a version of it up on the GEN-FISH website. I'm going to ask Paige if we can have it up potentially during lunch, if we can just password unprotect it, and if folks want to play around with it. We did show this to our end user committee. I ran through an example based on what I'm experiencing out in Cape Breton using eDNA based on Daniel. I don't know if it's a suggestion more than a direct dictate, but that's okay. But fine, yeah. And like, GEN-FISH is covering the cost. I'm like, oh. And so right now we still need some feedback whether or not, but it was developed and co-developed based on what fishery managers think important. So we're looking at the size of the site, how much money we have, the personnel, the access to it. Some of the questions that we knew were coming from our co-producers and users. The fish health toolkit, we thought at first going into naively, it could be set up the same way. We're just going to do a direct comparison of costs, of methodologies, of time on the water, of processing. And we realized that the fact that some of our end users didn't even know what transcriptomics was problematic. Right now, actually just yesterday, Holly, who's out in Cape Breton doing her research as part of our team, we always hold a community meeting, workshop meeting every single time we're visiting. And both at the beginning, and we keep everyone up to speed, but we have different sectors because our work in Cape Breton is fed directly by community needs. So we're always updating them. And this is the first time Holly was going to describe transcriptomics. And we're like, so we've got recreational anglers, we've got folks from salmon associations, we've got indigenous groups, we've got some DFO folks from inland fisheries. And we're like, okay, I don't have a script. Like, no one's prepared a script for us to know what is the best way to communicate transcriptomics and what these results are. I'm like, Holly, can you put up that little diagram from that first paper? But that's still not. So we're saying, okay, when genes change, I'm like, genes don't change. When genes get expressed, they're like, but express seems like it's such a semantics. I don't know, it's an actual scientific word. Genes express themselves. I'm like, so anyways, I'm going to get the update. It just happened yesterday. I'm going to see how it all went down and I will report if there's something that we could benefit from. But then again, when we're going to be doing it, it's going to probably be like a decision tree. And the first question we could start off with potentially, and this is something that Daniel and Ken and anyone else we're kind of like working through right now. It's what's the purpose? Is it for monitoring health? Is it for diagnosing something you think is wrong? Or is it to look for early warnings? And this is kind of what we're pitching to the community out in Cape Breton. It's both diagnostic and prognosis. And then the second one is, well, what are the alternatives? What are the conventional ones? Are we going to do a necropsy? So that's diagnostic. And are we going to do hormones instead? We're going to look at titers, concentrations, because that might be a little bit diagnostic. Well, what's monitoring? How do you monitor for health? How do you do it conventionally? So we haven't really gotten to exploring what these alternatives are. But then lethal versus non-lethal sampling has been something that's come through. Again, if we started this at the beginning, at the very beginning, GEN-FISH never would have been a question. Species taxonomic group, we can be more specific now at this stage because we know all of Ken's incredibly hard work, what we can offer with a certain amount of reliability. Cost effectiveness, we have to look again at RNA sequencing as an option versus the STP chips versus the traditional ones. The turnaround time for results is a big one. That's something that was mentioned for our eDNA end users. Others are already adopting the health toolkit. Do you know of others using it? Do you know of facilities that are out there? And if you actually have a known or a suspected stressor, that's also going to help us out. So not just a taxonomic group, because through Ken's work, what kind of stressor or multiple stressors do you think that they're facing? So again, still in production, but through no shade or fault of everyone, this just really does represent the cumulative knowledge and experience that we've all gone through, that we've shared with Activity 5. And that's it. I'm happy to take any questions you may have. Thanks, everybody. And lunch. Yes, Daniel.
Can you distill it down to what you think is perhaps the single greatest barrier for adoption of transcriptomics?
Knowledge. I don't even know what it is. And so you don't know what it is. You don't know what you should be asking or looking for. So the way we've gotten it to past our community members, and I'd love to hear anyone else that work with community members in terms of how you've pitched it. We're looking at, we can diagnose it before they start to die. That might be an overreach, right? I might be trying to sell it too hard. But then the second question is, before they die, you're going to kill it then to figure out? So then I catch myself in a catch-22, you know? So I'm like, we got mucus. So I think from my experience, it's that. It's not the cost. The cost comes later because they're like, oh, if you're proposing it, you're paying for it. It's whether or not is it understood, and does it seem like I'm trying to sell them something? Yeah. But what's your experience? Ken, Annalisa, anyone else who's used transcriptomics? Yeah.
There's my.
Yeah, right. Those online, please feel free to chime in as well. Ken, how about you? Like up in the north, are you trying to sell transcriptomics? at this point?
Most, like, yeah, in the north, it's mostly working with fishery notions. And so they're pretty much on board right from the start. And so it's less of a, trying to convince the general public or anybody about the need for it. I remember back when we started the Parks Canada stuff, which Bob Hanner and Margaret, it was once we sort of laid out what the approach was and what it's supposed to do, they seemed to be pretty excited by giving it a shot. So I think it's just, letting people know what tools are out there that are potentially going to be helpful for their system. Hopefully that leads to that type of buy.
Yeah.
And then, we had a different situation with those fish were going to be associated with already their fishing harvest. So it wasn't as big a concern about the legal standard.
Yeah.
I think it's a certain amount of trust as well.
Mm.
The Guinea Fisheries Authority, the senior fish biologist is a Thomas David. So he knew.
Nice.
Everyone did actually work with a number of people from the Guinea Fisheries Commission. They said, here's our problem. What can we do?
Mm.
And so pitched this idea.
Perfect.
So going to the people where they sense that there is that knowledge that we can actually produce something, then they have different interpretations.
Mm-hmm.
But it's going to answer a question.
I think you raise a really great point, Mark, because a lot of when the information comes in, bring it back to the community. So it's called collaborative analysis. It's a really well-known social science technique that sometimes we're kind of a little hesitant on because we're always seen as we're the conveyor of truth, whatever that may be, right? But it's more like, okay, what's your experience? How far back did it go? How long is this lineage? Did they go through a glacial event? Nations will know that through their oral histories. So getting that kind of information and asking for interpretation, I think none of us have mentioned that too, in terms of like, oh my God, the pressure's on us to try to write another Alliance grant, to do all these tests and everything. But the last step does come into this shared interpretation of results. Yeah.
That's what the case studies are just so valuable so that it's not just theoretical. It's just like, okay, in this situation, that you're able to use examples when you're talking about it rather than just,
100%.
The other issue is, education and communication is important, but sometimes it just surprises the heck out. So, when Corey thought it was an implantable chip, because the term chip is used widely in fisheries as like a pit tank.
Yeah.
Or people interpreting response to contaminants as being the fish are going to be poisonous if there's a strong contaminant gene response.
Yeah.
And which are natural, I guess, interpretations, but not something I would even think of somebody coming up with.
And you think about how it crosses language barriers as well, because in French, it's called a chip is called a puce, P-O-U-C-E. Please correct me. That's thumb for me, right? So like for me to even explain it, if I'm using the French terms, I'm doing STP, let me just call it in French, puce, and like that's still incomprehensible. And so if you're dealing across languages, the interpretation, like an interpreter, is really, really important too. So using acronyms like STP, that's why I like the health profile. Like visuals are so incredibly important to convey and get that information across. Yeah. Even when the data come back, visualize it. It doesn't matter what language you are speaking for anyone. For me, I'm a visual learner, so yeah. But there's something that we should actually record, or like, what are some of the barriers that we've experienced and challenged, but that's coming after now. Yeah. Major hands?
I'm just curious about what level, you don't want to insult your users, but what level of complexity do you need to get down to so that they have the knowledge to say, okay, I understand this tool. I mean, do you need to tell users that it's a chip that's got all of these genes on it? Like, you know, when we do screening for physiology, I don't need to tell our users that we're using 9612 plate that's lying with antibodies. I need to give say, like I've heard you describing your community members as yellows doing more or less. Like it's active, it's not active. Those are more easy to understand terms. I'm just wondering if there's ways that can sort of simplify the terminology to make it less. There's stuff in the word transcriptomics, right?
Yeah.
You have to use that word.
I have two answers for that. One is how already accepted is the techniques you're describing versus the level of trust. So if this is something new terminology, just think about CRISPR and gene editing. That opened up so many ethical concerns because people were misinterpreting it. So that knowledge had to happen. Now you don't really hear so much about gene editing. Now people are most of us are accepting of mRNA vaccines, but it has to be explained that it's not harmful. You're not injecting something necessarily. So I think it comes down to people go get their blood taken all the time and they're told about their cholesterol. So explaining a hormone you're doing to a fish, you don't have to overcome that barrier. So I think we have to be really cognizant of that. Margaret, you might shut that down. Go ahead.
No, I was just gonna say that's a really good point. I mean, what we're really talking about, I guess, like multi-marker, like you're doing able to assess multiple markers simultaneously. And I know I'm emphasizing the chip a lot. It's been when I've been writing for funding organizations to try to show them how this is so much more cost effective than doing a whole lot of RNA-seq. And so it's, okay, well, there's, so many, genes on a chip and so many samples and all that sort of stuff. And so then emphasizing the format, but ultimately what you're just talking about is able to assess multiple markers simultaneously, cost effectively, and whether it's a chip, whether it's this or that, doesn't probably matter to me. So it's very, yeah, and I think ultimately it's, who's our audience?
Yeah, exactly.
I think you have a really good point though. You don't want to treat your end user like idiot. You don't want to be, you want to have a lot of trust too, because you're not being transparent with what you're actually doing.
Yeah.
So it's this kind of like there's this balance point probably where there's a key tipping point where it's like, okay, don't alienate your audience by giving so much complexity, but you don't dumb it down so much that you don't have the tools to understand what you're actually doing with the tissues that you're taking from their community or their location.
Yeah.
Yeah, and I think then what it really comes down to is that it's a dialogue, not just that here's all of the information. I try to figure out just the right amount. That you sort of start with questions, and then there's more back and forth. And again, it's what you said about it's not so much an end user that we're delivering a final product there, but right from the beginning, that the back and forth. What are you interested in? How might this be useful? Well, we could do this, and that sort of thing. Using case studies to provide examples.
Yeah. Nick, you had a comment, question?
Yeah, I mean, think about it as if you go see the doctor, often if you get just a health check, right, they're going to say to the lab to take blood sample. You're okay with that. You don't necessarily they're going to measure that blood sample. The fact is that the lab is probably going to measure dozens and dozens of different things, come up with a profile that will tell you overall if you're healthy or not healthy. And it relates to your earlier question about what do these gene expression profiles tell us about performance, and we're not there yet. So we have to make the link to what the profile tells us in terms of performance of the animal. And it's just another tool to monitor performance. And so instead of calling it transcriptional profiling, it's just the same thing as you go see the doctor, you get a blood sample, and from your blood sample, you'll get a profile of a zillion different things. You don't have the knowledge to understand what this zillion thing is. That's the role of the doctor is to interpret that to tell you whether you're healthy or not. I see it that way, right? So eventually we'll get to what does transcriptional profiling mean in terms of the performance of the fish. In the end, that's what we're selling. This is the performance effective or not?
I think even to explain that, you know, if we're trying to find the drivers of it. Yeah, so yes, we're gonna be explaining, this is what means, it's under, this fish or the population is experiencing something. But if they don't have an idea, if the community or your partners don't have an idea of exactly what it is, this is when I think collaboratively information can be used to help figure that out. But you're right, the first you have to have a level of confidence to show this animal. But I like that as a panel, as a marker, just another toolkit to say the profile. How's everyone doing? Yeah.
So even we cannot, you know, human panels, even vet panels, you're not attacking, basically. What you get is, get where they are and whether they're below or above a certain level. And vets and doctors get excited when they those vets skills out of the mean. And so even so, I mean, with, you know, lessening the strain on us from trying to get performance and fitness, which is really hard to do. Even being able to say, okay, in this given environment, in the control situation when the fish is, lack of a better word happening, it's within this range. And when it's outside this range, this is most of the I think transporting kind of that idea of a panel and being outside of a certain range is maybe an easy way to kind of.
Just establishing what those ranges are. Now we're getting into fold changes.
Variability, you know,
Yeah.
Cross sites.
I know.
Too, but without having to say, we can only do this when we get to fitness and performance.
Yeah. Yeah.
No, that's good. That's actually a good stopping point. We can come back to it later. It's not for me.
Perfect. Thank you. Thank you.
Thank you. Okay, so for everyone in the room and online, I have 12:25 p.m. If we want to meet back here at 1:30, we can continue then. Okay.
So yes, we do get letters.
Okay, so I guess we're now at the moving forward part of the AGM. So we'll start with the fish survey toolkit. And I think we maybe touched on this a little bit yesterday when we were sort of thinking about lessons learned with GEN-FISH. And so now we're maybe thinking more specifically about moving forward, including, of course, you sort of wrapping things up and making sure that even beyond the end of June, we're continuing to make sure things get published and disseminated more broadly, and then potential for follow-up work. So who wants to get started?
Margaret, do you want to start with activity 1 or 2, or does it matter?
I would say, why don't we start with activity 2 this time?
Okay, sure. Well,
now that you ask.
Okay. So I do have some ideas. So the one thing that we were not able to complete was 2.1.3, which was looking at the abiotic and biotic correlates of the eDNA results. And I think there's an expectation that we will do that, and Sapna and Pedro will have first crack at that. And certainly if they only, if they I don't know, if they're no longer interested, there's others definitely interested. Obviously Matthew's very enthusiastic about it. I am as well, and in fact I have to write my discovery grant this year, and I think I will include this as one of the data sets that we will analyze in the future. So I think there's a lot of things that we can do with that 500 water body data set related to what we said we were going to do. And again, Sapna and Pedro will have first crack at that, and then we've already identified others who are interested, and as Matthew said, certainly open to people who are interested in collaborating on that. I do like the idea of the biogeographic analyses and particularly comparing conventional to eDNA. And that's where SAPNA's databases would come in really handy. So I think those are two areas that directly related to the 500 water body survey that there's some future there. In terms of the case studies, I think most of them are in various stages of publication from will be, thinking about a draft to already published. So I think we've actually done quite well with the case study publications, the primary publications related specifically to the case study, and then we're working on the synthesis now as well. And the thing about the synthesis paper, we did not want to steal anyone's thunder. And the synthesis paper is supposed to be have case studies that are, just short overviews of the case studies within a certain format that would if another paper is already published, would be cited as, to give the details of the case study, but then to map it against, the ways in which we could use environmental DNA in fisheries management in Canada. And I will point out that one of the case studies was with the MNR. Well, it was a case study and also part of the 500 water body. I guess a lot of the case studies are like that. But the MNR Northern Lakes project, for example, is we were very slow to get the results for that. But in that project, we actually paralleled eDNA sampling with the baseline monitoring sampling that the standardized baseline monitoring sampling that MNR does. And they have a formula for setting gill nets that is related to the size of the lake and the depth and so on. And it's a stratified random sample. We used exactly the same methods to determine where we're going to sample. We were going to sample eDNA on the exact same lakes that they subsequently sampled in the same year. So I think this is going to be a very strong database to do things like detection probability analyses and compare conventional to environmental DNA. And that is on my to-do list. I think I'm going to add that to my DG, and also Dak de Kirchhoff, who is the research scientist with the province who oversees the broad scale monitoring program. So he's very interested in being involved in that as well. And then one last thing, and I noticed Anne-Laure just put in a link to Thais's initial paper for our activity 2, and our second paper is going to be the bookend to it. But there has been quite a bit of interest shown by Parks Canada at the park level. So, for example, I was at eDNA and Lutsike a couple of weeks ago, reporting back to the community on our eDNA results. They were very interested in the results. They provided great feedback. We had high school students marking on maps where they find fishes, matching, and giving us the DNA names for the fishes that we detected. And it was very positively received by both the community as well as the Parks Canada staff. And because eDNA is a new national park reserve, Indigenous National Park Reserve, there's virtually no baseline information, and it's a massive area. So I think our initial 50 sites was a proof of concept that this could be used to develop baseline monitoring. And in fact, we've developed a Great Slave Lake chip to help with that. But the question is, how do we fund this going into the future, right? And we did have. I'll remind you that around 2022, I think it was 21-22, we did have discussions with Parks Canada about potentially developing an NSERC alliance to more formally develop and fund this, a relationship between GEN-FISH and Parks Canada. There was a lot of interest, but at the time there was no obvious way to mesh it together, find the required match, and so on. So we ended up working one-on-one with several parks, but I know there, Margaret worked with Wapusk, and we worked with Wood Buffalo. I know that I've talked to Point Pelee several times, and they're very keen on environmental DNA due to the lack of aquatic expertise and the challenges with invasive and endangered fishes at Point Pelee. Rouge National Urban Park is in my backyard at University of Toronto Scarborough, and there may be others. So my suggestion is, and I've already preemptively sent an email to Anne Lore after yesterday's discussion about reopening this discussion about the future of the use of environmental DNA to sample aquatic systems in national parks. So those are the things that I've been thinking about in terms of the activity 2 and the logical extensions to it. And I know Matthew's been thinking a lot about logical extensions as well. And I'd be happy to hear other thoughts that people may have about where to go next with activity 2.
I have some thoughts on that myself. So I work a lot in the relationship. So I'm always interested in kind of pushing what we can do with these technologies sort of a step further. I think we know now very well, it's widely accepted that eDNA is extremely useful for characterizing presence absence. I am very interested in the application of eDNA to do things like monitoring numerical abundance of biomass in organisms. I want to see how well we can translate quantitative eDNA data into quantitative estimates of abundance and biomass. And this is something actually it's interesting you mentioned Parks Canada, because my previous postdoc before I came to work on GEN-FISH was actually working with Parks Canada to do this experiment on the relationship between eDNA concentration and brook trout abundance in Banff. And I think that sort of for a GEN-FISH 2.0 or a Parks baseline study, a component that would be extremely interesting is the application of eDNA as a quantitative tool beyond just presence absence, but actually to look at things like abundance and biomass. And parks are really kind of ideal systems to work on these sorts of questions. There's one thing that came out of, like, we've got a paper that's finally coming out from that postdoc years later. The one thing that we highlight is that to establish these types of relation or utility of eDNA for these types of applications, you need baseline systems where you can essentially calibrate a model using things like metabolic data that you have in relationship between eDNA production and physiology, mass balance models where you look at the bathymetry, discharge, and input of water in lakes. And if you can set up in parks across Canada these types of baseline systems where you do the conventional work to establish the baselines, where you calibrate eDNA models, you have the potential to be able to start making quantitative interpretations of eDNA that you collect in other systems based off of the relationships that you can observe in these baseline systems. So that's something that I'm very interested in seeing moving forward. And I think if there is a component to a Parks Canada Alliance grant, that's something I think would be a very, very interesting way to move beyond just a sort of descriptive baseline of biodiversity in parks in Canada to actually push beyond that and then start doing those types of quantitative analysis.
So we have a couple from she says Chinese review paper that Nick was talking about is a great resource for the work that they're doing in Parks Canada. And she says not to make any commitment here, but she does think that Parks Canada is eager to be engaged in work as it aligns with our needs and mandates. Any kind of partnership should be constructed in line with fielding limits, needs, and capacity. And as Brooklyn says, to Matthew's point, moving towards analyses using digital PCR instead of qPCR for absolute quantification.
Yeah, I would just to follow up on and Lorraine and Matthew's points. Yeah, we really, I think the key is like we've done all along is to use co-production to identify what our partners' needs are, and then how we could help address those needs with in eDNA. So I think it's critical that we have a discussion that is based in co-production of identifying needs and potential solutions to those needs.
Yeah, and I'm just jump in because I think we've got these are really exciting ideas for sort of like the next big grant, and what I've been thinking about a little bit more is maybe possible sort of bridge funding to sort of help finish up some of the GEN-FISH 1 deliverables, because we're already losing HQP, and I say we're losing them. They're moving on to bigger and better things, but I mean the number of master's theses that have been produced, but maybe the paper hasn't yet been written, and it's always very demanding, almost unfair to be asking, graduate students, et cetera, that are no longer getting paid for a project to still continue to produce papers. And I dread the end of June when when Chelsea's officially done, but there's still a lot of additional work. So even sort of thinking about some small like bridge funding that can help continue to support HQP as we continue to finish up a lot of the GEN-FISH deliverables. I know we all have the will to do it, but sometimes having the ability to do it is another thing. And so I was wondering about, like with some of the things, whether there's, like I was just thinking, like with Matthew, when you were talking this morning about, number of how many sites do you need to sample in a given water body based on the size. I mean, sometimes while we're also working on the more academic publications, guidance documents that would be useful to various partners. And if there's opportunities for small contracts for some of those things that would be of interest to partners. I know some of the work that I'm doing with various partners where we're doing eDNA work for, species X, species Y, including money in the budget to help support the HQP to also finish up publications on various things. And so some of those things I think might be really useful sort of bridge funding as we start to plan maybe the next big project.
Yeah, Margaret, I'll just follow up on that. Yeah, the bridge funding is necessary because, as academics, it's not like we have a big pot of money just waiting there to be used. And the way I've been thinking about it is I'll be using my next discovery grant will be some of that bridge funding, right? Well, having students working on focused questions. And, I think, you brought up the, how many sites do you have to sample in a water body, and, in the secondary question, where and what depths and so on. To me, that's like near future questions, because they're essential to optimizing the use of eDNA, right? If we don't have that guidance, then we lose some of that expected efficiency in taking this approach. Of course, the other challenge will be for those of us who actually don't have our own genetics labs, and this is maybe sort of related to the commercialization, because if some of us academics don't have them, certainly many of the partners will not either. But it sounded like that there will be an ongoing possibility for fee for service for some items through University of Windsor. And I hope they keep that up, and I hope that I understood that correctly, because I think that will be a really important element of moving forward and getting broader buy-in.
So Nick, to that point, the EGF has been set up with the idea of doing both research as well as fee for service, and Shelby's been pursuing that. And I think the more we can run projects through the EGF, the more stable the future the EGF will become.
Mhm.
So
Makes sense.
I think, it all comes down to funding.
So when I get consultants saying, where can I send the eDNA samples? I send them to University of Windsor, I think.
Yes, Shelby's done contract work for consultants as well as government, as well as research.
And the middle of a couple right now.
Yeah.
Yeah, because I would guess that most of us who do some eDNA stuff, that we would be able to do single species assays if it's particularly if it's ones that we do ourselves. But yeah, anything, the open array chips and metabarcoding, yeah, that's definitely EGF. And I know, I mean, even at University of Manitoba, sort of exploring this a little bit, if I do cost recovery, I can only recover the cost of supplies. I can't do salaries. And as we know, it's kind of the salaries that are really critical. But yeah, no, good to know that, particularly if there's enough consistency of incoming contracts that you can plan accordingly, then that's that's the whole sort of point of the EGF.
It seems like, if we do work on things like bridge funding, it would be good to include Windsor in some of those discussions to sort of help with that planning and costing, and also to make sure they just don't end up with things out of the blue, or we send something 3 years from now, then we find out, oh, they went under because they didn't have enough work or whatever, right? So sounds like Windsor should be looped into those discussions, and we plan on using that facility.
Something I'd like to bring up in terms of future is the comparison of the open array versus metabarcoding. Similar, to what Kyle was describing with stomach content DNA and eDNA to identify fish communities. What is the relative contribution of metabarcoding versus the open array? And we'll have that data, but we're not going to be able to analyze it. And I think that would be a low-hanging fruit that would generate a lot of potential support. And actually refute the value of the open array eDNA. And I think it'd be an easy analysis, particularly in the northern lakes. If we have that data, if we know what the fish community is, the problem is always the gold standard. And that's why the goldfish ponds were so cool, because we know exactly.
Yeah. And I would say, if we're doing something like that, I think what would be really useful too is also to add like an explicit cost comparison too, because I know that as academics, we tend to compare things from just sort of from the academic output point of view without doing a really explicit cost. And I know that it's going to vary. There's no, single number for each of the two methods, and that's necessarily going to be applicable 2 years from now. But how many times do you read that that eDNA is more cost effective than conventional sampling, and there's not a single number or even a sort of an estimate of time for the different methods. And so that is best as possible. And even if it's just like for this study, running the samples through the EGF, this is what the time was for each method, and the cost to again use it sort of as a guidance document for end users as well.
I can't even hear you.
Oh, sorry, everyone. I'll speak up.
Or at least project.
Okay, so Anne Morse says, this is a huge concern for Parks Canada, since we don't have any molecular lab within the agency nor affiliated lab, and every park contracts with their local community or academic partner. We ended up with several ways and protocols across the country.
Yeah, well, that sounds like an opportunity, right? And I look forward to discussing that opportunity further with Anne-Laure and also any interested GEN-FISH partners and PIs. We have standardized ways of doing it, right? That's one of the strengths of GEN-FISH. We've been doing it in a standardized way. So I look forward to those discussions.
It also goes beyond Parks Canada. If DFO is the outlier, and to a certain extent ONRF because of Chris Wilson, but there are a lot of government agencies that are interested but don't have the lab facilities and don't want to put that in, that investment in.
Case in Manitoba, they're really keen to sort of partner with us if that's an option, because yeah, they wouldn't be running it themselves, but they would be willing to pay to have samples run. And a lot of our collaborations and a lot of the species at risk funding we get is from DFO, that the DFO lab in Winnipeg is at capacity already. And then one of the things that they find is that they can't scale up quickly, like especially in terms of hiring new personnel versus at a university. It's just like, oh, okay, you know what? Let me see if there's a student that's interested in working in the lab for the summer, and turns out there are. And so it's we can respond very quickly to sort of influxes of funding, so that even with DFO, yeah, that there's the interest. Oh yeah, Brooklyn, go ahead.
Yeah. Hi, everyone. Sorry, I'm in the lab right now. I'm actually in Margaret's lab right now, still. So yeah, I think all of these points are something that I've kind of experienced now, being more outside of the GEN-FISH world and outside of the fish world as well, because terrestrial eDNA and non-fish aquatic eDNA is what I'm delving into now. So there are different sources of, I guess, like, there are different opportunities available. For example, like I work with ECCC now a lot, and Nature Conservancy of Canada. But yeah, one of the contracts that I got last year at the place that I'm at we're an NGO, was from ECCC looking at mud puppies, which are aquatic, of course. And they originally had a contract with Karen Helbing's lab, and they were working with them all the way over there because they have the fee per reaction or fee per sample option. And that's something that now I'm starting to develop in our little lab so that we can have contracts like that. I know it's more difficult with academic sources to be able to have a contract, although I'm not really sure how the chemistry departments at the U of M is able to have a privatized company that does eDNA. But regardless, what I've seen is definitely a desire from various organizations to have a fee per service that they can contract you to do. So I think that's something that needs to be explored for sure. And broader too, because sometimes, like there is obviously the facility at Glear, but sometimes people need the answers within, a certain time period, and to be able to build capacity in those facilities as well, to have more staff who can, help you out as well, or more consistency in staff is also a big point that I think needs to be addressed.
Yeah, and I would say that, yeah, that if I wanted to and I thought there was a need, I'm sure I could set up something at University of Manitoba. I mean, I wouldn't really, I have no desire to do that if it makes more sense for stuff to go to EGF. If you're at capacity and what somebody wants to do is, Carmine Shiner or native lamprey eDNA assays, then maybe that's something that it's at University of Manitoba. But yeah, wanting to make sure that the facilities are available to end users, but also making sure that if there's a dedicated facility at University of Windsor, that potential contracts aren't spread so thinly that it ends up that EGF isn't viable. But yeah, and I guess so I'm not sure if this is where we talk about potential commercialization. Like I get I mean, with the idea, especially with the individual assays, either single qPCR or open array, that at least with the single species assays, we're obligated through Genome Canada to share them broadly, so that if there's consulting companies that think that there's a market for some of the particularly popular species, that they have all of the information they need to do that themselves, which still means, though, that we're obligated through Genome Canada to make sure that everything is packaged up and shared fully. I mean, at minimum on a website, ideally in a publication as well. Yeah, so that I don't know if I don't know enough about open array to know if there's any sort of commercialization options there.
I think we're going to talk about that a bit more on the.
Yeah, that's okay. That's what I was.
Legacy.
Yeah.
In terms, I think the bridge financing, I had not thought of that. I think that's pretty critical because there's a bunch of things that are just so close but not there.
Yeah.
And it's just not something that people can pick up and do on the side of the ledger.
Yeah.
I know I've had success with both DFO and OMNRF getting small amounts of targeted money.
Yeah.
When I say small, like 20,000, that kind of money. And if you could say, this is what we're going to do, it might fit in. And if they just have money in a budget sitting. But that's a very sort of one-off kind of thing. But if we have a target, then maybe we can start shopping it around.
Yeah, and I think if we have, several small pots of money one-off, that they add up, because I know if all of the work that has been done in my lab depends on me to wrap it up, it's not going to get done. That I can't do it without the HQP, and I can't ask the HQP to do it without funding. And so, so being able to tap into those pots of money, again, fairly good with getting, as you say, those small pots of money for specific projects. I tend to often under budget stuff, like this is what it will actually cost, so therefore that's exactly what I'm going to ask for, instead of, padding a little bit, not outrageously, but certainly to plan for, not every single PCR working, the first time. I mean, like, how many times do we budget for best case scenario, which never happens? So budgeting for those eventualities, and also, budgeting for time to actually wrap things up, not just run the last sample and we're done, but even if it's not a publication, a report that you're sharing with the funding agency. So yeah, making sure that we're thinking of those things. And I think, I mean, we've had 2 Great Lakes Fishery Commission Science Transfer Program projects, whether there's one or 2 small things here that look like they're appropriate for science transfer program. Again, really trying to make sure that things that are like 95% done, if we need a small pot of money to get it finished up, and that'll definitely, I mean, make us all feel better. We won't be lying in bed at night feeling that, okay, there are those 3 things that I still have to finish off. But of course, then it'll really position us well then if we're looking for a large pot of money for, phase 2 or something like that.
Margaret, that reminds me of, yesterday we were talking a little bit about science transfer, just to do a final report back to our partners and collaborators, right? And so we should certainly consider doing that.
Okay, good. Yeah, because I thought that it had come up, but I couldn't remember the context. Yeah, thanks.
Yeah.
activity 1 is great interest for Parks Canada. Would the qPCR assays database be evergreen or somehow updated at some point? Considering new species will appear in some areas because of climate change and habitat shifts or introduction, and putative new hybridization, and then playing with sensitivity and sensibility of qPCR assays.
Yeah, and that's a really good point. And again, making sure that we're being realistic and we're in our planning and what it will actually take. It's one thing to just, okay, we're done GEN-FISH, we're going to put the spreadsheet up there, but if we want to maintain it and update it, then we'll have to make sure we commit the resources for that, because I hadn't really thought of that, but it will become obsolete if we don't maintain it.
You could make it a living document, sort of like NCBI.
Yeah.
Where people can submit new assets to be included in that database. And it's already big and impressive enough that it might become the go-to place if you're thinking of doing eDNA work and you don't already have your own asset.
Who maintains that data set and updates and does like
I've got you.
the cleaning and all that?
Yeah.
Right? Perfect.
Yeah.
Checks it too.
Couple of, sorry if I'm talking over people. I can't hear people in the room. Sorry. Go ahead and just let me know when I can talk.
You can talk now, Nick. Go ahead.
Okay, sorry. A couple of things come to mind. One is, the government of Canada is going towards open standards and open data standards, and is Genome Canada not part of the government of Canada? And then the second thing is, there is this University of Saskatchewan project about, or Regina project about orphan data sets, and not to say that ours is orphan, but that is a group of experts who think about how we deal with long-term maintenance of data sets. So it's someone we could actually contact just for advice even. Bruno Soares is, I believe, is leading that. But that they may have some ideas and potential solutions for helping with long-term maintenance of the database.
Yeah, that's a good idea. Yeah, because I mean, it'd be great if others could contribute to it so that we're not responsible for also, those last, 10% of the assays that still need to be species specific or finished. If others are contributing to that, it adds the extra complication of, curating it and whether do people upload stuff directly and then we have to, check what they've uploaded, or is it submitted and then it's checked over? And that those are, and again, I guess everything that we're talking about, we're recognizing that any continuation, it's going to take resources, and we need to budget accordingly. And I don't mean necessarily always just budget money, but budget time to make sure that we're being realistic in what can be done and how to do it. Okay. Very good.
Yeah. Yeah. Some ideas. Canada. Yeah. Was the database Canada Computes?
Digital Alliance.
Yeah. Change the name. Yeah. So
What's next?
The obvious thing for activity 3 and 4 is interpretation. And we already sort of got into that this morning. And there was an initiative to start an alliance grant. We got to the point where OMNRF, a couple of consulting companies, DFO, the BC Freshwater Fish Society had all agreed to contribute in kind and possibly cash to an alliance proposal. And then I got busy and it didn't go anywhere. I've been in discussion with Nicolas Bernier about him being the lead. I don't mind working on that proposal, but I don't want to be the lead of a 3 or 4 year grant now that I'm retired. So, and I already stiffed this guy as a junior early career researcher by making him the head of an activity. So the idea is
opportunities.
That's opportunities. That's the way to think of it. So in terms of sort of a grand hopeful, next step for activity 3 and 4 is that strategic to come up with software that will interpret patterns. So more than gene by gene, actually looking for, as discussed this morning, you starve the fish and then you thermally challenge them. The profile will let you know that they're nutritionally stressed, and that might tie into software that would say, okay, that will reduce the response to other stressors. It's like, you know, handling stress reduces immune response. So.
And then the strength of the ability of the program to actually be able to pull out those patterns is going to come from all the data sets that are going to keep flooding in over the next little while too. So then also, well, I think we originally talked about doing a lot of data mine or data downloads of various RNA-seq papers as well to try to get, you know, an overall transcriptional pattern that's associated with particular responses.
And the intention of that would be to also have some natural science where targeted challenges would be included. The scope would be narrowed to salmonids was the idea.
Yeah.
Just, you know, I'm sure people are interested in other families, orders, but we've got to have it more focused than GEN-FISH was, because we're not going to get $9 million in our alliance grant.
And did you say we'd also be looking at other papers outside of GEN-FISH with all their transcriptional data as well to add?
Yeah,
it's not just confined within GEN-FISH, but it's all that transcriptional data floating around.
Yeah, just to beef up the amount of data that would go into any type of analysis. But I think I mean, I don't understand how this stuff works, and I'm not the person who should be commenting on it. But yeah, I think if we have, you know, whole transcriptome patterns that demonstrate a response, and we have chip data that kind of was applied to the same types of stressors, that might help strengthen the ability to, you know, tease apart those patterns using the chip data.
Nicola?
So we've started to discuss about how we're gonna basically put this data in public domain. But because we at least activity 3 and 4 never came up with a standardized protocol of how we analyze things, and all kind of develop our own ways. While it'd be nice to take advantage of publicly available transcriptional data sets, unless you've got the raw data, and then you can compare apples to apples. In terms of the analysis, it's a bit complicated, but at least among ourselves, we have to come up with a way before long, before all the issues are gone. Have the raw data sets.
Yep.
Again, what I mean by raw data sets is actually, you know, relative fluorescence.
Yeah. Yeah. What's the file called, Shelby? Yeah. The nice thing about GEN-FISH is all our data is coming off the OpenArray platform. So between your machine and Shelby's machine, we can download all of those and get comparable data.
And that would be the most powerful thing. And yes, we should take advantage of other data sets. I think it gets a little more complicated.
The other issue with other data sets is we picked our 112 favorite genes. And if it's on unrelated function or even related function, but different gene, then we don't have all the assays that people have tried. Like within GEN-FISH, those assays have been tried on a lot of different fish under a lot of different stressors in lots of replicates. So, but I think that where other data would come in handy are the transcriptomes. So applied stressor before and after. Shelby, you had your hand up?
Okay, I was gonna comment on, because I know earlier we talked about how like if a fish is getting stressed to the point where it's kind of giving up, that might be an issue. But I was just gonna comment on like a lot of this is for monitoring, so we should in theory be catching the fish when they're actually responding and not that 12-degree difference in temperature where we saw the first fish kind of take out.
Mm-hmm. Yeah.
So it should.
Absolutely, and that's what's the good response, or what's the terminology, the eustress versus distress response in a fish.
So that would require metadata for everyone's study.
Yeah.
You know, it would be like time of day, ordinal day, because we just need so many drivers.
Mm-hmm.
It's so responsive to covariance in addition to the treatment.
Mm-hmm.
Even Ellie's was showing that she wasn't expecting a sex difference because they're technically juveniles, right?
Yeah.
Ellie's, I don't want to miss the fancy. But so those kind of things too, that would add noise, that would obscure the patterns that would emerge otherwise. So we'd have to set that up as part of the alliance, is like, let's set up a metadata base first.
Yeah.
Or alongside it.
Yeah, and that's critical to being able to do anything with the EDS files in the future, is to have that really nice curated metadata, which includes the version of the chip that was used and which primers for that gene were we using for that particular study. Sir? Yeah.
Mm-hmm. It will quickly balloon into a very complex metadata sheet for that analysis, and the least of our problems will be the qPCR data. That's going to be the walk in the park data. It's going to be the metadata that's going to be the challenge.
Yeah.
But needed, did you say?
Totally. Yeah.
And sorry.
But I was just going to say, I suspect the first round of analysis of the metadata is going to identify gaps. And that's why my view of this grant or this proposal would include people who are going to take salmon and say, okay, we don't know what will happen in terms of immune challenge, because we didn't do a lot of immune challenges in GEN-FISH. So there might be targeted experiments either to verify the function of the software or to add to the data, because the metadata is just not there. Nobody thought to record the time of day, and yet the circadian rhythm gene's going off the clock, off the wall, and you're thinking, how is this possible?
Just to carry on with what you said, it's also about how the data get analyzed as well, because I remember when his lens submitted her paper to CJFAS, she analyzed the data one way, and the reviewers came back and said you have to analyze it a separate way. So I don't even know if we settled on a common way to analyze the data coming out, whether I'm using CT or you're using not, we're going to LinRay. I learned that from Daniel, thank you very much. But I don't even know if we have a standardized way of analyzing, because I do know there are different ways.
Yeah, and one of the ways that Yeah, absolutely. And one of the ways that we've been doing it is using it's a Bayesian approach called MCMC QPCR. And so, yeah, it was not that it's the best, but it was a really quick way of being able to incorporate all the efficiencies for all the different assays really rapidly into the analysis.
And as Annalise was saying, it's using endogenous controls as the prior.
Yeah.
Which helps with those sample size, correct?
Yeah, I guess. Still, you can still do it without priors, but then you've got your electrical for wider. I will say, like, even within my own studies, I was like thought between using MCMC PCR and just the like negative and negative delta CT. So it depends on like the question I want to answer. Like for my segmented models, I use like delta CTs because I want like my already taken into account. So yeah, even just me,
That highlights a really good point.
Yeah.
Yeah, and I think that any attempt to try to come up with a standardized interpretation mechanism is going to have to explore the best analysis.
Yeah.
Because the reason why we use LinReg and backtrack to estimated original concentration is then you can take the undefined and say that's a 0, as opposed to if it's undefined and you've got CT values of 32, 34, 35 and a half, and then undefined, it's lost data. But in fact, it's just that it's below detection limit of the assay. As long as the amplification curve isn't insane. But yeah, no, it's a really good point.
End user, co-users requirements too, right? I mean, if you're producing data for an academic, they want something they can analyze as raw data. If you're producing something for a community, they might want to know whether there are particular stream or rivers in the bed zone. They don't need numbers, they need to know whether they need to do same about remediation, right? So like there's different outcomes for different users, and not one of them is like that red zone is useless to an academic, doesn't tell them anything. But for community, they don't want those specific numbers, doesn't tell them what they need to know.
An interesting point, and Tina keeps pointing out the difference between activity 1, 2, and 3 and 4. The academic users, all they want are the assays. They don't care because they may not use OpenArray. They may use a different platform, or they may be interested in just 3 genes. They just need the primer and probe and you know the PCR conditions, and that's all they want. The non-academic user is a huge hurdle because the data is so technical. And that's what I think we're talking about developing is the non-academic. And I don't like that because if you're an ecologist, a muddy boots ecologist, you might be really interested in using this tool to determine whether your, brook trout population is stressed. You have no idea what cDNA is, much less how to interpret CT values. So that's the type of user that we're targeting with this.
You're also targeting me as an end user, someone who doesn't understand the technicalities, collect samples, send it to a team. I need raw data back, or at least data that you tell me I should put in the stats model. I don't know how to run assays, don't want to run assays. I don't want to know how the assays work. I trust you to basically get them done properly. So there's an in-between one too, where I'm an academic, or I might be DMO or whatever, and I'm a scientist. I'm trained as a scientist, but I want raw data I can understand.
You also need to analyze. So Daniel gonna give you.
Starting at CT value. Yeah.
And
there's a diversity of like mega academics, like it's a 10 part of it. We're in a lab and understand everything. And then there's in between this academic wants these tools, but doesn't really have to use them. But you collect the data really well. You tell me, like Margaret, you said, you've got your nice sheet like exactly how to do things, controls and things like that. And then there's the community, or even a government visitor that doesn't want numbers. They want to know, if I tell them, that this is green or red, right?
So then it's gonna be keep the line, you take notes?
So I mean, alliances are meant to bring everyone together, but I mean, this is almost sounding like a commercialization of these tools. It's kind of what this is, commercialization is the wrong word, but it is taking these tools and putting them into practice, so end users can actually use them.
And that was the goal of activity 3 and 4 for GEN-FISH. And we didn't get there.
Yeah, 4 years.
Yeah. I believe that the alliance goal, and this is sort of strategy, is to develop something that's going to make this technology useful for consultants and government and NGO and First Nation fisheries and et cetera, et cetera. But I think we're going to do it would involve a lot of science.
Yeah.
And there will always be applications where, Tina and I collaborate, you do all the hard work. And I say, okay, this is how you extract messenger RNA and make cDNA. Those are collaborations. But then whoever the client is, you're the client in this case, is dependent upon the molecular lab. And in many cases, the user may be suspicious of the molecular lab. So if DFO takes up on this and decides to do STP-chip analysis and say, your fish are fine, then the question is, it's a black box. But if a fisheries council decides we're going to do it ourselves, we're going to take samples, we're going to ship them to Shelby, and she's going to send us the data, we put it in Nick and Ken's Shiny app, and it pops up saying, okay, your fish are thermally stressed. Then they have control. They have ownership of it.
Is there anything comparable for transcriptomics at this point? Do you know of?
The one that sort of I thought was really well done was the ecotox chip through the University of Saskatchewan. So they have a website where you can take your output from the qPCR machine, and then it is a GUI based web site that you can then do the analysis yourself. So it's plug and play, so you lose a bit of your ability to really see what's going on, but you can even download example data sets and run it through that program and get an output as well. I don't know, well, it's a little bit more, we're all friends here. It's a little more straightforward with toxicology because, certain pathways are really linked to specific types of contaminants. But I think, yeah, I think they might have some ability to even try to interpret the results as well. So I thought that was a good way of making this, the analysis kind of available to the public.
And I'm so sorry, I don't mean to dominate. You mentioned yesterday, was DFO had something called EcoFit, or it was
the FIT chips?
FIT chips, Christine Miller.
Yes.
Is that anything that's similar to what we're wanting to do as well?
I don't believe that they have some sort of, it's not necessarily available to the public, and they don't have some sort of analysis platform that's open to everyone. I could be very wrong there, but I don't think so.
The one that's in Regina, sorry, what's that one?
Saskatchewan, ecotox chip. But that one's only for fathead minnows.
Model organism.
Yeah, boring.
So then I think that GEN-FISH 2 should have the 500 water body project for gene transcriptional profile.
Jeez.
Yeah.
I can't help but notice everyone refers to GEN-FISH 1 and GEN-FISH 2.
But it has always appealed to me to compare transcriptional profiles among populations within a species.
Mhm.
I think that would be really cool. The logistic, what's the word? Tough part is, unlike eDNA, you go and sample the lake and you're done. You have to catch the fish, and I really believe you've got to do a response to a standardized stressor. Because if you go in the lake and you grab it and the fish might just be having a bad day, right? It just was chased by an invasive predator or whatever, and so it's got a totally anomalous transcriptional profile. But if you go and you say, okay, here's my first of 500 lakes, and you collect the fish, you let them sit for 24 hours, then you have a standardized challenge. I'm thinking probably temperature, but I don't know. And then just look at before and after, and then move on to lake 2 and do the same thing. It sounds crazy, but there have been some physiological fish studies that are just mind blowing in terms of the scope. And this is where they did very similar, but then they measured cortisol. You know, a bunch of us know those, and they're just massive studies. So that's sort of a real long-term future direction for
another one of activities for GEN-FISH too. You know, because you could do something like use the experimental lakes area where they've been monitoring abiotic factors for decades, and then grab the fish from those isolated lakes within there. And
yeah, that would be perfect. Yeah.
We're looking at intervention adaptive potential, adaptive capacity. We actually have that in our grants.
Yeah.
That's one of the, not really deliverable, but one of the ways we can look at how much variation exists within a population, amongst populations, you know, across the geographic, biogeographic range. Would that be something that would still be of interest towards management? Because say whether this population, are there individuals in this population who have the ability to respond? So don't worry, or everyone in this population is responding the same way, whether or not it's in one direction or just, you know, very limited. Or that's just way too far advanced.
I think this gets back to Oliver's comment about how do you interpret a transcriptional profile. And I remember getting into arguments with Brian Dixon, who decided not to come to this. And I told him how disappointed I was. But if a fish is upregulating immune function genes, is that adaptive or is that maladaptive? If you're really well adapted and a pathogen runs into you, you get rid of it before it even becomes systemic. You never upregulate your specific immune system. But on the other hand, you might say having upregulation means you're fighting it off effectively. So that is definitely adaptive. So you could argue either way. Have the same problem. If it goes up and then down, that where it's crashing, but the stuff that Patricia has shown with her stuff, is that sign of transcriptional dysregulation and the fish is dying, essentially. We never got to that level, I don't think. I think individual studies did, but not as a network.
I think for freshwater fishing can from January it's 2 degrees Celsius, or it's the last week of August, it's 25 on the lake. It means it's only good.
Yeah. That gets back to the metadata for the information that has been generated by GEN-FISH being so important. But I don't think they're, unlike activity 1 and 2, I don't think there's sort of any stuff that desperately needs to be done that's sort of hanging there. You know, there are some chips that need to be refined, but they will probably be refined under individual labs discovery grants. A lot of the challenge, like the things that aren't complete for activity 1 and 2 are sort of beyond a single person's discovery grant. So I think there's sort of a difference in the future steps. I don't think we need bridge financing. I'm sure you'd like some.
Yeah, if we're applying for it.
I'm thinking of the hasn't been printed, which chips haven't been printed yet?
Eel.
Then there's that native one in Northern, yeah, in Northern Brooklyn. Oh, you thought you were gonna get away. Look.
We placed the order. We, yeah, go into the specifics, but we were trying to, lamprey chip version 2 is going to be more applicable to Northern Brook lamprey. And that was factored into the assay design. No.
Okay, yeah, I did pull away from the recorders.
What? Did you get the email?
It's a great exhibition.
Mhm.
Out of curiosity, we don't have an American to be named this.
A what data set?
We don't have any samples from American.
Yeah, I was, when we were talking about printing, whether we were going to put the American eel chip on the roster to get it printed and use up some of the budget, I contacted a bunch of American eel people and they had samples that they would ship that were suitable for RNA extraction, but it wasn't a challenge. It was like juvenile glass eels from 4 populations, that kind of thing. And I figured to get it all printed without a single challenge just wasn't worth it.
But it is there for future consideration.
Yeah. And as I said, there was considerable interest amongst the individuals I contacted about they'd be happy to challenge, but the timing wasn't right. We were talking about doing this the last 3 months. So.
Bridge funding might be needed for something like that, or design for something like that. American eel will be, I think, up for reassessment soon by CASI.
Yeah.
So if there could be potentially some species at risk funding or something like that.
Well, Scott Pavey said, send him the money. He'd be happy to do it. Good. I wanted to bring something up. I'm putting you totally on the spot, Shelby. GEN-FISH requires, Genome Canada requires that we put data to be publicly accessible. To be responsible, we have to provide enough metadata and description so that people, it's not just a data dump on some website. And we're working on that through Pedro and SAPNA. But curating is another thing entirely. And it struck me, how about if we create that eDNA website and put you in charge of curating it? And then if people want to send a new qPCR assay, you know how to look at it and say, wait a minute, this melting temperature between the primer and the probe is different by 20 degrees, so forget it, bozo. That's it. So
Don't worry.
You can quote me, but think about it. And won't, but that to me sort of would fit into potentially your role as providing a fee for service. I don't see, maybe I'm wrong, but I wouldn't see getting hundreds of submissions.
Yeah, it's pretty limited field, so it wouldn't be crazy.
Yeah.
Yeah.
Get lots of spam.
So, sorry, I forgot to ask a question. Guys, basically. So we moved in this grant to Mucus, which is really cool. Wasn't originally. Can I just ask, what are future ideas about working on blood transcriptomics? Is that a dead end, or is that still something that's of interest? And what, you know, in the humanists, what are the limitations of going blood? We didn't do it this one, but is there any interest?
And did you do blood transcriptomes?
I haven't done it myself, but I've worked closely with someone who did it, and I guess they're just a pain for the extraction part. That was one reason why I've kind of shied away from it. But there's no reason not to try to develop that for sure.
Well, Patricia did a blood transcriptome for her undergraduate thesis. Do you have that data?
I do. It's not. You have like raw blood transcriptome data.
And plus liver and gill for the same fish.
For the same fish, yes. I will say that the blood mRNA transcript counts was much lower than that we feel liver, which was to be expected. But it was exciting that we were still able to see this RNA from blood.
Yeah.
Yeah. So we have the data.
I mean, I guess if you've got Lucas, then, and it's giving you great correlations to Gill, then why would you want blood? So
Or it's blood or blood, what processes, what blood, because the gill and these are both external basic organs. Blood would reflect more metabolic, internal, maybe gill again. Don't know.
Yeah, I mean, it'll definitely pick up a lot of sort of aerobic performance type measures, and then immune response stuff as well.
Look at that data again. I know we gave up on it because we were going to test for the actual challenge. But if we just compare blood to liver to in general.
I mean, for me, it was more of an interest, you know, as a physiologist with the blood cell base. But now that you've got Lucas chip online, that many cases surpassed. And you kind of need to do something with blood.
That's true. You hormone guys just throw the blood cells out. Well, birds are good too because they're nucleated red blood cells.
So it might be a moot point, you know, because now that the nucleic acid kills work really well, less invasive than needed blood. It's just not. I was just curious.
It'd be very different genes, I think, that you detect.
It might be very limited too.
Okay, I think we're off the hook. Thanks.
Hey then, so for the last one, the decision guiding toolkit, it's these toolkits, this decision matrix that allows you to figure out whether or not eDNA can and should be employed alongside conventional methods. And the other one is like, what are transcriptomics? Should I use that to be able to diagnose health? In terms of the progress, I don't see it really going anywhere. I know that sounds really dour, but even if we're getting feedback from our end users, for us to manipulate it and add more questions, it depends on, because Peter, who worked on it, I stalked him because he wasn't getting back to us. So he's now working in an engineering firm and he's just not responding to any of our queries. So that's one of the downfalls of when you're finishing a project and the money dries up, people move on and they're no longer reachable. He did leave us his HTML code, but again, it's going to take someone else to pick up that mantle and like dig into that past the longevity. So how much that is going to be a tool that we can manipulate on the GEN-FISH website, it depends on who's interested. So I was just giving like the bad news in terms of where I see it going. In terms of the transcriptomics one with this decision matrix, I can see some longevity if we tie it into the GLFC science transfer program kind of grant that we have. But again, unless someone's willing to take up that mantle, maybe as we can weave it into that alliance in terms of not only are we going to explain what the results are, but we're going to also attach to that what transcriptomics is, how it can be used, and what are some of these considerations. So in terms of that kind of longevity of these tools. So again, just laying it out there how I feel. I feel like I'm like Nick Bernier, a little bit more glass half empty than glass half, or cautiously cynical.
Just plain cynical.
Yeah. Don't want to overpromise, right? That's the worst thing you can do. It's just a promise and then not follow through. So I don't want to make empty promises here. But the whole point of activity 5 and of GEN-FISH altogether, what can we do to make these tools adoptable? So I see us kind of folding this into the legacy and the commercialization. But before we even get to that practicalities, how do we get the word out about these tools that can be provided? When we hit up our end user committee, I mentioned it kind of yesterday. They mentioned, and this is like our feedback, what we can add to it was to attend the Indigenous Great Lakes Network. There's an annual meeting. Show up, talk about it, talk about both tools. And you're going to find there's going to be a difference in the people's perception. So we almost need a way to, how do we explain? Oliver, you had that question this morning. How much depth should we get into? Where do we start? And I think it might just be a little bit of just reading your audience and who that might be. Other groups or other ways is LinkedIn. So we heard that back from our end user members. Find out, because a lot of the times some of our end users were saying that they find out about a post, like events and training through LinkedIn. So if there's anything we do, if there's anything new to our website, look, there's this new metadata. I don't actually know. I'm not on LinkedIn. I have to ask other people who have an account to look. But is that possible to do on LinkedIn, to post your own events and trainings and then tag people? Right? I'm crossing my platforms here. So yeah, there's that. There is something called equivalent to nature metrics. Some have said, like live demonstrations, methodologies, and protocols. So again, follow up on that if it's something we want to do, or if it's something that your lab is doing that you're interested in. Meet with your community members, meet with your partners. I don't know what a live demonstration would look like with transcriptomics, except for maybe swabbing or anything like that. But everyone likes to take a picture of the chip. But that's really like, we'll invite ourselves to your lab, shall we? Yes, pass it around, right? Webinars, virtual road shows was also recommended. Again, this is all gonna be like, who's gonna do this? On whose behalf? This is no longer GEN-FISH. So it might be on behalf of your own, how you want to get it out there. And so some of us might feel like we don't have there's no onus on us to promote something unless we're getting a grant and this is actually part of our research program. We're not going to do it selflessly necessarily. DFO has a community of practice, and there are been talks before potentially on conservation genomics. And so if you have a partner already in DFO, ask them about their community of practice seminars, whether or not you can get in there. So again, because I think the take home here is that a lot of our HQP will start working at DFO, at Parks Canada, all across the different sectors. And so by doing this, I hope we have some really good suggestions too. By doing this, we're ingratiating and we're spreading the knowledge, and potentially we're like, oh, here's an HQP who has these skills already. This is really important. You might want to hire them as well. GLFC, so it's the Great Lakes Fishery Commission. They also have a technoscience seminar series. So that's somewhere else. If you're looking for money, join it so that you can promote these different tools and say, hey, guess what? I actually have experience in that. So that's, I'm open to hear about other methods of adopting both these kind of techniques. And a lot of it's word of mouth. So if you think about it, how did you, sell these techniques for your partners? eDNA is much more common now. It's a lot easier. We've got folks approaching us to know. Right now I have my students just telling the community, like, my lab can do eDNA. I'm like, I am not an eDNA lab. I know people because like, oh, we can do it. We know there's GEN-FISH. And then they went on the website and they saw GEN-FISH is ending. So I just got a text going, GEN-FISH is ending. These are non-GEN-FISH students. I'm like, yeah, what's going to happen? I'm like, that's what we're talking about on Saturday. Okay, so can you read me out if you don't? Oh, yeah, the comments, or go ahead, Paige. Oh, that's nice.
Ann Moore says, we have just created our community of practice of conservation genomics, including eDNA approaches within Parks Canada as well.
Oh, nice. Good. Thanks, Anne-Laure.
Says like a month ago.
Oh, nice and recent. Sorry, Anne-Laure, would there be room for transcriptomics for that kind of meeting, or is it too soon? Maybe too soon. Oh, yeah. Okay. So Anne-Laure, what do you think about introducing transcriptomics as a tool, or would you rather us wait until the alliance is much more developed, or in the terms of the confidence that you might have in just introducing it? Is in it as well. Yeah, perfect. Nice.
So in a way, just send an invitation.
Yeah, send an invitation to Daniel and Ken, you're off the hook. You've done too much. Yeah.
I've just been slacking.
Mhm. I'm sorry, Paige, you wanted to say something.
Yeah, just.
Instead of focusing so much on the project of GEN-FISH, focus on what GEN-FISH has developed. So we've been referring to all the toolkits. This conversation with you earlier today, of like toolkits kind of implies you get a physical product, you get like a 23andMe kit or something. That's not really what we've developed. We've developed like data sets, information with the STP-chips. It's like a whole new mode of testing. So even doing something like changing the name of the toolkits might make it easier to find and more immediately understood what it's about. So like Aubrey said, like fish health panel instead of fish health toolkit. Or we have the fish survey database rather than fish survey toolkit. So we're not sending out the samplers anymore or anything like that. And then these decision guiding rules are tools. So I think that's actually a good thing. But something like that, to focus on what our deliverables are at the end of the day and making the website reflect that rather than this is a Genome Canada grant.
Right.
Does that make sense?
So sorry, instead of the fish tool survey toolkit, it would be,
Fish survey.
yeah.
That's it.
About toolkits team resources, I guess now GEN-FISH moved on from testing these things, actually give them product use, which is database instructions, protocols.
Yeah.
We're just adding the products, right? This is what GEN-FISH was, this is what it developed, now this is what we produce.
Yeah.
Nice.
If I may just ask a quick question, the domain of the website, who owns it? Who's paying for it? Who's upkeeping it past June 30th?
I'm not sure.
Great.
It's part of our mandate to keep it up for 10 years after the project ends. I don't know how technically that will occur, but that is what we've promised to do.
Yeah.
But I don't know through what money.
That sounded like my cue.
Yeah.
I was listening. We currently have the domain for 3 years.
3 years. So I'm just letting the folks online know. So the Fishcast, the GEN-FISH website domain is going until 2028.
Yes.
Great. Okay. Maybe 2027 late. Okay. Yeah. So again, when we're talking about, yeah. Yeah.
That's what our discussion is focused on. And I know it's part of the legacy. I know it's the commercialization we want to interweave in, but it's also the legacy of how it's going to keep going the momentum. If we don't have this like bridge funding, new grants, part of our NSERC discoveries, smaller like GLFC grants. But Margaret, you had your hand up. What did you want to say?
eDNA sampling protocol and eDNA assay database, something that's very descriptive within that, so that we aren't changing something that maybe people are familiar with, but at the same time, having something within that that's very explicit. Yeah, that makes sense.
Yeah.
But I would actually, I tend to agree with Paige. Those names were almost imposed on us by Ontario Genome. Because at the time they said, what the F is environmental DNA? And nobody's ever heard of transcriptomics. And it needs to be a widget because it's Genome Canada. So that's why that fairly uninformative terminology was developed. And, you know, now pretty much everybody who might be interested knows what environmental DNA is. Transcriptomics is still a problem. So the fish health assessment or something.
Yeah.
Yeah, just check my email. The domain was renewed late October 2024 for 3 years.
Thank you. So October 2027. Nice.
So on biological ecosystems is one of their top research priorities.
Nice. Okay, good.
That makes sense because we've got Gerard from Parks Canada who wants us, Parks Canada, to do some transcriptomic profile.
Yeah. Which fish species?
Shark.
Sure.
So I guess there's
Very cool.
some place of monitoring up in better sort of animal populations, monitoring them for perjury and other persistence.
So I'll use you as a case study then. So you're Gerard, what's his name? Sorry.
Gerard.
Right. And so then did he know about transcriptomics beforehand? Or did you bring it to him? Or he knew of you, obviously he's working with you. So how did that knowledge exchange happen or transfer happen? Oh.
So Patricia, all Patricia's fault.
Patricia gave a top that was looking down that
Was it?
Yellow, the guy who's excited by yellow picture.
Absolutely, yeah, as I got approached
At
by
the conference.
But was curious, like, as to how he could like send samples to the EGF and potentially use our chip for these purposes also. He wanted to know about the application of his chip or of our chip with his work.
And I think he does a lot of work with Bob Hanner, so I think he's, you know, firmly,
He's in the
very
full.
enthusiastic already about
Yeah.
the fish thing.
So that's an interesting point. You mentioned that yesterday at conferences, you've been approached about the technique and the methodology. And I do know that SCAS, it's government heavy. And I know that one of some the ways we're trying to improve SCAS's outreach, this conference, scientific conference, is that they don't have a lot of ENGOs or even industry. And that's something that we've been working on the board of SCAS to try to promote more and more, simply because these NGOs don't think that there's anything that they can get out of that. And in terms of even networking, they've got established their own networking. And can this government academic do they even have a place in it? So there are those of us who are on the boards of different scientific communities or government, because you're heavily involved with GLFC. I know Steve's heavily involved. So there's still a way, if it's still part of a research program, just to make other sectors. And again, Nick with Markel, working with the SON. So again, this is why a lot of it is just boots on the ground kind of thing. But if there's more innovative ways and techniques, it'd be great to share them. But again, it takes people who want to, it's still part of the research program. Yeah.
SCAS would
Yeah. Oh, that's right.
Trying to position ourselves well for the alliance for GEN-FISH 2.0, that might be a good sort of launching.
Yeah, fantastic. Yeah, Matthew.
Also, just want to put a plug in there since we're talking about conferences that Anna was talking to me about trying to organize another crises, which is a pathway to increase standards for competency in science. I got that right, hopefully. But it is an eDNA focused conference, and he wants to get that set up in 2026. And then we'll learn all the other crises conferences. There was a substantial NGO and private sector interest.
Nice.
I did comment yesterday in the call that it is going forward next year.
Mm. Are there any genomic kind of networks and outreach that we can do that's not part of scientific conferences that we know of? Or our own? Should we have, is there a genomics? Oh, I guess it's genomics. Then it gets muddied with other things.
The issue is generally genomics conference. Highly technical. The idea of actually applying genomics to solve real problems is front of them.
Mm-hmm.
That's pejorative.
No mention of CRISPR then. I know. Okay, so part of it is obviously adopting it and being convinced that it can work. Then the second part is show me the money, right? Commercialization. Who's going to do it? So I'm gonna step down off the podiums and hopefully it segued nicely into legacy / commercialization. And that's another way of promoting those who can adopt our tools that we could come ready with, because those are some of the questions I'm sure we get. Can you do this for us? What do we need to do? So I just want to make a plug again for the work that Paige and Margaret have done in terms of, I really like that with the GLFC, how you went through that checklist. So the first is like, is eDNA comparable or transcriptomics, yes or no? Then how did you collect it? What's the checklist? And I can see that potentially emerging from a potential alliance as we get our standards up, because that's practically what that was, right? A standards checklist, pre-flight checklist. I really liked how that was done with a report at the end. I think that's incredible. And if that can continue that momentum with the transcriptomics, I think we're heading in the right direction. And then that's that interpretability, like interpretation at the very end. That's a big piece that collectively we really need to work on. I do see them as being distinct. So I'm going to say once again, we need our standards. Then we need to get those standards out in a reporting form. And then we need to also say, like who's going to do it and what the cost is going to be. And those all have to be interwoven together. So that's all I'm going to say as I then pass it on. Is it back just to you, Daniel, or everyone collectively? Paige? It's casual? Everyone. Okay. Then we're talking seriously about then commercialization. Who can do it? How much the chips cost? Is that it?
Yeah.
Okay, perfect. All right. Thank you.
Hey, Trevor, are you actually there? Can you turn on your mic?
I'm here, yeah.
Can you give an update on the commercialization?
Yeah, no problem. So I was listening in carefully. So the commercialization, I'll give a couple general comments because I happen to run the commercialization for the university at the moment, and I have in the past. So two challenges that large grants like this face are, one, faculty honestly are not great at commercialization. They walk away from projects at the end, and we have lots of commercializable things at the University of Windsor, and only the engineers seem to actually pick up and run with it. So that was one of the challenges. So it seems like science is not generally good at this. So we're trying to change that culture. So, and I don't know about other universities, but that may or may not be the case. The second challenge that we face specifically for GEN-FISH that we need to avoid in the future is you can't have an IP agreement with 16 different universities because navigating that, we talked to the lawyers more recently, is just a nightmare. So part of it is that nobody has ownership because you've got IP going across the entire group. And so it's really tricky. So in the future, we talked to some venture capital people briefly, and they suggested that the way to fund this kind of thing is to parse off something midstream and have a dedicated group to work on it, not have this tragedy of the commons where nobody focuses on it. So I think those are the two big challenges. One, that we're not very good at it, to be frank, as scientists generally, because that's not our passion. And so we either have to farm it out to another team. And the second part is setting up the IP agreements more carefully at the start, because right now we just have this very generic, almost unusable IP agreement across the universities. And to split any kind of financial issue, which has come up several times when Tina spoke and others about, where's the money in quotes, it makes it financially not viable for the universities to run with it. The last thing I will say to you, that we've dealt with this a little bit, is that commercialization with genetics, a lot of you know this with genetics specifically, but for those that don't know this, it's very hard to patent things related to genetics because you can't patent many aspects of it. It's more the process or the product and not obviously the genes themselves or the outcomes from the genes. So I think there needs to be much more forethought into the IP if we go for a GEN-FISH 2 is what I would say. And we should have brought in, if I could go back in time, we should have brought a IP specialist on board, even if it cost us $20,000. So we should bury this into the next grant. We should put $20,000 to $50,000 in year one just to bring an IP team on to set us up for success. And I've started to do that with other people right now at the University of Windsor for engineering, where we're bringing people in at the start of a big grant and saying, how do we lay this out instead of retroactively doing it? So I know that was a lot at once, but I apologize. But that's sort of the big takes I got from GEN-FISH.
So Trevor, I get the impression that we missed the boat on true commercialization.
I don't think we missed the true boat. I think that the chip, and I'm using the chip loosely here, is still very viable. What's not really viable is the IP agreement we have across the universities. So we'd have to revise that agreement before anybody would even touch this right now. So when we sent this IP agreement on before Heather left, they said, what is this? This is untenable. How did this even happen? So we're getting kind of retrospective advice that we really didn't set up the commercialization very well because we lacked the experience, frankly. So again, it's not all doom and gloom. It's not just, what did Nick call it? Cautious skepticism. Doomsday for Tina. So I think we just learned some valuable lessons here that really inform us for possible GEN-FISH 2, where we really need to actually dedicate some funding to IP development in the grant, because really we were doing it off of the side of our table, off the corner, and it's not really viable. So I'll just leave it there unless people have questions about it. But the agreement we set up just wasn't tenable to really run forward with any kind of. We made $50,000 to be split 16 ways, and the cost of the patent be more than all that. So we just didn't set ourselves up for success the way it was.
So at the time, Trevor, we were just desperate to get the grant up and running.
I understand.
And so we went with a boilerplate that did not consider the possibility of 16 universities.
Yeah, so my main point is not to criticize what we did, as opposed to saying let's do something different if we do a GEN-FISH 2 and really think forward on it and bring some IP people in and from the get-go to help us.
Okay, now my feeling would be is if the University of Windsor was to reach out to the other universities to renegotiate the IP, at least if I was another university, I'd immediately think, okay, wait a minute, they actually found something valuable. I'm not going to give up the IP from University of Manitoba, for example. Do you think it would be possible to renegotiate IP at this point?
When they read the IP agreement we have, it's so broadly classified, they said that it was functionally useless, but as you suggested, it's any other university would be foolish to give up their rights. So I think with the chip itself, we could do an among PI kind of thing and say, who really worked on this chip? I didn't work on it, for example. I use it as an end user. So I would say, well, I don't need to be part of that. So maybe there's a way to, within our own group, do something like that before we get to the university level.
Okay, so it's not a closed door, it's just.
No, I will tell you also that I'll give you an example for Windsor. When I did the review of intellectual property on campus for 2007 to 2019, that's the ballpark of the range, we spent $1.78 million on patent costs and made $30,000 total in profit. So there's the ratio of cost to profit that Windsor has had over more than a decade. So I would say that other universities also have this issue, but they kind of bury their costs almost as if it's a lottery ticket where, you know, Western and Waterloo dedicate serious funds to it. And they get the odd hit that actually makes them money, but Windsor has not had the success to date, so they've been reluctant to really invest heavily.
So, given that we're at the end of GEN-FISH,
yes,
I would say the chances of any of us taking on the effort to deal with this IP nightmare are low.
Yes, my suggestion, if I had the the big wand in my hand and I could magically do what I wanted, I would suggest that there should be some discussions about a small spin-off that is separate, and then work on the chip, for example, with Fisher, as we did have some interest, and of course it got disrupted with Heather's sudden departure, not her choosing of departure, unfortunately. But yeah, that would be it. You need a dedicated group. I mean, it's just everybody had their piece to work on scientifically, and we all did our thing. But the commercialization didn't have a champion, I'll say. So if we can try to remedy that in the future, that would be really wise.
Okay, I think this is an action item that doesn't have to be tied, because commercialization is not required by Genome Canada or Ontario Genome. They'd like it, but it's not a requirement. But maybe this could be sort of an action item. Perhaps Ken, you and I can reach out, you can say, do you feel you contributed to the development of the chips? I think the eDNA chips are a non-starter. We have to release the assays as part of the Genome Canada requirement, and then just printing them on a chip is not really all that innovative. But the work that we did on potentially validating the STP-chip, maybe this alliance grant may generate something that's actually commercialized.
Yeah, if I can interject one last time, because I don't want to belabor the point, there's something called IPON, IPON, which is the Intellectual Property Strategy of Ontario that does fund efforts like this, just so you know. So each province has a fund separate that you can apply to for funds. So if you had a champion for this, there's certainly opportunity for a completely separate set of funds. I heard people talk about Great Lakes Fisheries Commission, other opportunities, but there is a specific IP granting portfolio that you can apply to. And in Ontario, it just happens to be called IPON.
Okay.
I will also finish off by saying that the other thing too is that if, you know, we're really fixated on IP patents, things like that, but more, if we're talking dollars, the real money here would be to have someone generate service and interest for sending things to EGF. So if we had almost like, you know, Shelby, that's not her job, she's a technician, but if we had someone, for example, that we had on the books that is on commission almost, that go find all this work for GEN-FISH related products in the future or this time, they could easily pay off their own salary. So that was suggested to me by some VC standing for venture capital fund people, suggested that you need a person that's being paid to constantly be at it, looking for clientele and looking for services to be sent to wherever, EGF or whatever hub we wish. So that was the last comment I got from some VC people.
And Trevor, do you think that Windsor would have any appetite for supporting that kind of position?
Not its current fiscal situation, but I know there's IPON money that we could apply for through the VPR. Yeah, so that money could help pay for the person, but it would be another grant writing. So this is another challenge that we all have, is just a grant fatigue, of course. But I don't, in the current fiscal environment, you might be aware at Windsor, I doubt there'll be money sloshing around. But we do have staff that work on these things, but not dedicated. So we do need to find money for a dedicated person in the future.
Hey, Dan.
Yep.
I was half listening in and hiding up in the other room, but it's related to this. If the GEN-FISH 2.0 involves Genome Canada, I would strongly suggest that the lead be outside of Ontario. Not because I don't want to be involved, but you can appreciate the administrative burden on this project has been stifling. Financial reporting every 3 months on a $9.5 million project is insane. The ROC reports, the progress reports, It's actually a miracle we got anything done, because all we were doing was reporting. So, you know, I think Mark, you can speak to this, because somebody, Paige was telling me, look, BC, some of the genome stuff is, that kind of scrutiny isn't there. It's more, they actually want the progress to move forward.
That's one question. It hasn't been a problem in BC.
I was asked to participate in the BC Genome, and they said, fine, but you can't have any money. So they get my CV and then know that we can't transfer BC Genome money outside of BC. Because BC Genome actually puts a lot of money and effort into the Genome Canada projects. I think when we talk about GEN-FISH 2.0, our tongue is firmly planted in our cheek. Nobody here wants to lead another Genome Canada project anytime soon. So I don't think that's an issue. It's just even like the strategic grant, not strategic alliance grant, to me, that's sort of GEN-FISH 2.0. Yeah. Moving on. Right, Genome Canada just was not designed for this kind of project.
No, it was painful.
Mm-hmm. Yeah. Yes. Okay, thanks, Jeremy.
So, to buy this team some time to get to the alliance, their NSERC has an idea to innovation set of grants. Has anyone ever heard of those before?
Heard of them?
I've heard them and I've reviewed for them. It's been a few years. Yeah.
So they're designed to accelerate the pre-competitive development of promising technology originating from the University of College sector and can promote its transfer to a new or established Canadian company. This is achieved through defined phases by providing crucial assistance in the early stages of technology validation and market connection. And partner eligibility is actually pretty wide. So it doesn't have to be a specific company. It could even be a university technology transfer corporation or a university incubator. So I wonder if this would buy you some time to kind of, think about wrapping up some of these products, getting them ready for commercialization before you get them to launch them to like what GEN-FISH 2.0 would look like. And it's just looked at by NSERC staff. And there's 4 submission dates per year. One of them is June 20th. Oh yeah, but one of them is September 22nd.
We can do it.
It's like a month's going to come. I said it's the same ambition there, guys. Yeah.
I mean, it doesn't say that the budget looks pretty open, and it's not designed for sending conferences. It's not designed for training students. It's specifically designed to sit down and figure out how you take your technology and get it to someone to use. So it might be useful in the interim. That might some amount of short-term money to kind of not let GEN-FISH kind of fall on the wayside. There's also, keep talking, there's also synergy grant. So I mean, Trevor and you, Daniel, went forward for Dial a number of years ago. I mean, like when you read the description for synergy grants, I mean, it's this team approach. Synergy awards and honor outstanding collaborative R&D teams who have combined their expertise and resources to create partnerships resulting in significant impacts for Canada and Canadians. I mean, you could consider doing a synergy award too, just to kind of like, but you and Trevor have more experience with that, of course. And Nick, you were involved in that as well. And Mark, you were involved in that too.
Yeah, Ollie, we've done the here at Windsor before, in engineering. Again, you need a champion who's willing to run that through the system. That ideas grant, they call I2I in short, is really meant for someone that's in that valley of, they've got a product thinking into that valley where essentially things disappear. So it does exist. It's an incredibly onerous process, to be clear, but it does exist. But you need a champion, again, someone that's leading that effort. And so that's always been the challenge, to have one single individual with a singular purpose of going after the IP piece. So we really need an IP champion, is what I would argue.
For me, like looking at all this, it's almost like the next thing is to kind of take the EGF and turn it into like almost another company. Rather than intellectual property from the chips you've all developed, it's almost like the next stage is, okay, how do we publicize this to the rest of Canada so government organizations and contract companies can come to you and basically use the expertise you've developed all the way from the chip, all the way to providing the product.
I would see that as a need. And, whether it be EGF or startup. The problem with a startup is then you have to have the capital to buy the infrastructure, and you have to have somebody who knows how to run it without a big learning curve. So EGF would make a lot of sense. What are your thoughts?
I mean, I'll take any business. Your retirement makes me nervous.
Yeah.
I gives you an EGF Instagram. Time to the clear one.
How do you, and are you allowed to, this is a very naive question as an academic, how do you commercialize something like EGF? Are you allowed to?
He's part of this. I feel like it's already kind of commercialized in that sense, which is on their websites and advertised there, right?
Lots of models, Virgin all the centers in Canada. Allow it's like a class of example, it's to sit there. Use so much business. And you're already service.
Yeah, because we already have a
Both
lot.
internally and externally.
Yeah.
And to my mind, whatever we do, if it becomes a burden, then we shot ourselves in the foot. There has to be some benefit, because it's going to incur cost, whether to one or more of us, or at least to you, Shelby. But I see if the EGF is doing this on fee for service, I would probably, help solidify your position and help keep the EGF open for academic applications. So I think it's something that's worth trying for, because I see the benefits, not just the costs. And in fact, at the ROC, the Research Oversight Committee commented that heading over IP to Thermo Fisher is dumb, that we should keep it and get some kind of benefit from the past 5.5 years of work.
Yeah, I think, Daniel, that the idea is not to hand over the IP, but to license is what really makes the money. When you can have a licensing agreement with a company, that's what you want, not selling the IP or giving the IP away.
So that they actually pay us every time they sell a STP-chip?
Correct, yeah. So that's the royalties off of the licensing deal. And that's the $30,000 in all of U-Windsor history, that was from a licensing deal we had with a car company. So most universities only make money off of not selling their patents, but actually licensing access to a product or software or whatever. So I think licensing is what the ROC was alluding to when they said that to you.
Okay.
And we're doing that, the cost of the chips is going to be driven up, and then the likelihood of people using it might go down. Because GEN-FISH gets a hefty discount.
Okay.
Is that gonna continue?
I get a discount, not as much as GEN-FISH. GEN-FISH got 75%. Yeah.
But it comes back to like what all of you were discussing, which is like, comparing seining versus electrofishing. If you could do some kind of market analysis where, you talk to your partners, and you've already probably talked about these costs, what it would take to do a normal survey of seining and identifying the fish of human hours and salaries and all that, and you're able to say, I could do this with eDNA with less sampling, with more accuracy, more precision at the end of the day.
That's what the toolkit is.
That's
That's Peter and John's.
I mean, it's just there, and you have a sellable product online that even if the cost is a lot more than what you all would pay for, you've got something that's sellable. I mean, it's not like all of you don't already know this. I'm not saying anything you don't already know, just
So I don't think the purpose of this is to come up with action items specifically, but I think this is something we need to talk to, first of all, Thermo Fisher and see if they'd be willing to do a licensing agreement. And if it's, 22 cents per order of chips, which is 1,000, then I'm not too worried about it. But if it would actually generate real money, then I can see that going maybe into the EGM. That's natural. Is that the cost? Any more thoughts on commercialization? I think, Trevor, you put it well one time, we had a meeting, is that we're academics and we're not good at this stuff. Nor are we attracted to doing it. So I'm like, wow, that sounds like fun. I want to learn IP law.
Yeah, it's accurate. I think the real challenge is there are companies out there that you can actually pay a licensing fee to go chase down this kind of stuff. So there's a couple of engineers on campus at Windsor that have done this now, where you pay a company, they chase down the companies, they deal with the license agreement, and then they come back to you with that. And then you essentially work with them. Of course, there's a cost. But again, the only time it's worked is when you have a dedicated champion. No company's going to fork over money to you if you don't have consistent contact and consistent interactions and more sales and more people interested. And you have to do your part. A lot of faculty, often when I discuss it with them, they think, oh, I did my job. I invented X, Y, and Z gadget. It's your job as the university to find me money. And it just doesn't work like that in the real world. So again, we either hire a postdoc or a grad student, for a year or two to try to chase down business for EGF, or there's different models, but we certainly haven't had time to explore that during this iteration of GEN-FISH while we're all trying to get our studies done and get the students trained up.
Yeah.
And if we wanted money, we'd all be working for Fisher, by the way.
Okay. Good. Anything else to add to commercialization?
Maybe I have more of a question. So from my understanding, so it's not so much that we're going to have a product to convert a lot, but maybe the best opportunity for some sort of income in the future is offering services using the tools that we developed. So yeah, whether it's metabarcoding, eDNA, or transcriptomics, the chips or individual assays that there, I suspect there's enough people out there who would happily send samples and have them run rather than having to figure out how to do it for themselves. Setting up some sort of a system, if that was something we were interested in doing, to be able to supplement ongoing work, then that might be an option.
And also, as Trevor was saying, possibility of a licensing agreement with the thermal fission. But the problem with that is going to be somebody's going to have to convince them they're going to make money.
Yeah.
And we don't know.
And if it's unlikely to make us or the university much money and it's a lot of work, then it's probably not worth our.
Yes.
If it's not what we're interested in, if we're interested in making sure that the EGF can keep going, or if some few individual labs are able to support HPP by running assays, then that's a different thing rather than seeing it as some sort of a money-making venture.
Yeah. So I propose that maybe a couple of us meet with Shelby and Trevor and talk about a strategy that might be the best way forward, even if, we do generate money for service contract. Okay. Moving on. One of the requirements for GEN-FISH is that we do archive our data in a publicly available format. And we did have that data meeting where Pedro was saying that there were two possibilities. One was Dryad, but Dryad would pay for them to archive the data. And the other one was?
It was Compute Canada.
Compute Canada. right? Which is sort of, that sounded most promising. And the other possibility that we've been talking to
The runoff, or not the runoff, sorry, they're going to see in the future.
Yeah.
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 The competition. Yeah, the attorney was basically Canadian. Sorry, man, I was screaming about it 3 times. Yeah, basically, I'm talking to Roger Bull, who's kind of manages a lot of their biodiversity sort of archives and very interested in promoting inclusion in eDNA as the standard sort of a standard component of biodiversity archives for museum specimen collections. So basically, all of the eDNA being left over and the filters are going to the Roger, including any metadata and database that we produce. So there will be a whole work there as well, at least for the component of it. Yeah, so I think that is, as far as I understand, basically going ahead, like I'm going to spend some time this summer, check the trap stuff, sorting through our samples, and kind of organizing them, ship them to Roger, and then they'll do whatever they do.
That's a done deal then.
Pretty much, yeah. I think so.
Okay, so we can say that eDNA samples and sequence data can be provided to the Canadian Museum of Nature.
Yeah.
And they will archive it.
Keep it indefinitely. Yeah.
And because it's a museum, publicly funded museum, it will be publicly available.
I don't know. I assume it would be because it doesn't public funded.
Yeah.
But I'm not sure. Like, can you go on and just kind of access their archive samples now? I'm not sure how that works. Or like if they have plans to make that stuff. I don't know.
It's interesting that sequence of data as well.
Oh, they'd be taking the database and I think the raw files, so probably nothing,
So just like the terms and files.
Exactly, terms and files, and then the database is like cleaned and manipulated and matched metadata associated with the signatures, but there is a lot in between those phases, but over the two. Exactly.
And that's basically clear for one trying to get the university off my back, but okay, to stay.
So this makes me happy because independent of what they do with it, it is archived on a publicly available database. May have to provide a request to the museum, access it, but it's still available.
Yeah, that's probably like you guys say, anytime you share it, like you don't want it.
And it is publicly available or can be shared publicly if we need to have an embargo period before it's released because we're wanting to publish it. No, I'm sure all of that can be.
Or certainly one of the
Yes. That's what I was thinking. Yeah.
papers to publish that. So
That sort of information will be needed for the final.
Yeah.
And Genome Canada understands embargoes, but it can't be, it's under an indefinite embargo, it has to be this.
Yeah.
Yeah. Good. Then the STP-chip, we've got the transcriptomes. We just don't have an NCBI, right?
Yeah, that's the best place.
Yeah, don't delete the files in case it.
Yeah.
Mails and CBI.
I was just thinking, so with some of our northern work, DFO partners with the St. Lawrence Global Observatory.
Okay.
And that's a curated, publicly available data repository that can be linked with papers. And yeah, I just thought that now maybe that could be a place that could be useful for storing some of the actual chip projects. And then, I mean, I haven't talked to them about it as an option, but it's that's kind of what it's designed to do. They say that they're just an open access web platform making available to all data, information, and knowledge necessary to understand and effectively manage the ecosystem of the St. Lawrence from the Greater Eastern Gulf. And then they've also incurred to do other work associated with the coastal baseline monitoring programs. What I like about it is that they've got a full-time staff to help with the uploading and the curation, and we do environments as well.
But like some of BC species, they wouldn't, the target in that reach.
They've expanded out from it.
Yeah.
So we send our stuff there.
Okay.
What's it called here?
The St. Lawrence Global Observatory. Yeah, I haven't talked to them about this, it's just on my thought about chat.
Okay, so for eDNA, stuff's gonna go to the Canadian Museum of Nature. The list of assays that Margaret's in spearheading. So that you're going to curate that. And use it even out as promotional. And then RNA-seq can check data, give what we can. Maybe all of them.
That'd be cool, because the problem with the Digital Alliance is that it's kind of just most servers. It still requires some level of knowing how to access them and everything. So it's not, I don't see it as being a super user-friendly data repository for anyone wanting to grab the data.
Yeah, there are two issues for facing. One is the requirement from Genome Canada. The other is actually making it functional.
Yeah.
And it'd be ideal to do both.
Right.
But in the short term, we just need to be able to say, yes, it's our document, and trust me, it will not be there in 10 years.
Yes. So yes, I'm doing is it just the eDNA ones, right? Not the STPs, that's the configuration.
At this point, it would just be the eDNA assets. Because if we do the alliance, the STPs data might be valuable for that.
Everything we're talking about so far, well, I guess except for some EI is sort of basic Canada, because I'm not sure if that matters, and we could potentially matter if we had all of our data housed in USGS or something like that, and then all of it gets.
I feel that NCBI, I'm not sure it's going to continue.
I know, yeah, because that's, yeah, it's kind of it's the standard, kind of where that kind of data goes, but yes, it just does make it feel more comfortable with all of the other stuff that we're talking about, make sure. Sea lamprey, not sea lamprey, I see that now, if you get sea lamprey, sorry, St. Lawrence, sorry, but yeah.
Now, in terms of data security, Sharpie, could we get duplicated external hard drives?
Yes.
New data. And then you can keep it, in a drawer and yes. And then if whatever platform we've got the data on, it's simply disappears. We can't access it. That's not probably available, but it's secure. Because data security is also.
So one of the things that we discussed with you, we had a meeting a few weeks back, is the fact that a lot of the stuff's already published, and there's a lot of the eDNA work is already published. Already been deposited in a database for publication purposes, it's already linked. You don't necessarily link to the Museum of Nature, and now you would have two links to the same data. That can be a problem.
For the eDNA, most of the papers that are published are generated from the case studies, which are kind of their own independent things. So there'll be some level of duplication and nesting essentially with those available data sets as they, like, you publish Ian's goldfish paper, we're going to have to put on the goldfish sequencing data. And goldfish ponds are part of the 501 survey, but the 501 survey broadly is a much larger project that has its own sampling components to it, so I don't see there being much of a problem in terms of classification component there. I don't think that's going to be an issue for the eDNA side of the data repositories.
Yeah, I don't know exactly the intricacies of those things. Some journals like publish that. You don't have a choice. It has to be dried. A lot. Increasingly, journals have made arrangements with Dryad. This is where they tell you this is where the data is going to be. Is it okay to have it there and to have it somewhere else? No idea how that works. But at that meeting, I got pretty excited about data unification. Yeah, and essentially wasting global memory storage type thing. I would say it's not our problem, as long as we don't duplicate files, for instance. But if it's going to be on NCBI and Dryad, you'll see that being involved. Or with the observatory, previous observatory, it's there, sort of dedicated, and then Dryad for the individual papers, so it's scattered. As opposed to this.
I think that everybody worried about the education, but I just given the state of the world and the current problem we're discussing, having redundancy in long-term data storage, well, even if it might be wasteful resources, it doesn't seem wasteful to me. Absolutely. It's an insurance policy. I agree. Yeah.
Well, it's not like we're done counting. You know, we're not getting devil's value because we're depositing into these two spots. Hey, that's two publications. My CED's looking better. It's just redundancy. It's governance.
Yeah, yeah.
Padding our back.
Is Trevor still on? So he's going, Trevor's going.
Yeah, and I think especially like, at least for the eDNA database, I mean, that's kind of the summary. And so then there might be sequences that were generated to be able to design an assay, and so that for publication of a particular case study, that we might need to put the sequences into the GenBank and have a GenBank accession number, and then the GenBank accession number goes into the spreadsheet so that they're cross-linked without like duplication, because I know like this phylogenetic stuff, but it drives me crazy. I've seen where, you know, one museum has shared a subsample of their tissue with another museum, and then that subsample of tissue gets entered into their museum with a new accession number. And so we've got one sample that started out being a little, no one was sure it was identified correctly. And it's just like, oh, well, there's 3 separate museums that all showed exactly the same thing, and it turns out it's all just a subsample that was the same thing. Yeah, so that, yeah, making sure that we're not, you know, double counting and double numbering, all that sort of stuff, but rather making sure that they're all nicely linked together so that you can track. Yeah.
So unrelated and related, that question is, you brought up the fact that both you and Nick said that all data from the 500 water body, you'll be able to mine this for years to come. We're thinking along the same lines of the data from the chip in terms of being the raw data potentially for a PhD students in bioinformatics. Would you want to be in that position where everything's been now available, publicly available? And like Nick is gonna, like Mandrake said, he's gonna incorporate this in this next discovery grade, or we've got a PhD student whose entire PhD depends on data sets that are not publicly available. I don't know how you navigate that.
I think embargoes would be answer. Embargoes.
That's 2 years.
Yeah, you could probably, I think you could probably stretch it, especially if you get an argument, like say Nick has it like written up in his discovery grant that he has awarded or something like that. I'm like, okay, I have a PhD student who's gonna look at that as a 4-year term. I think you could probably argue something like that to get a bit of an extension on that. But I think, yeah, there is a block.
Yeah, I think, you know, if you were to submit all of this to St. Lawrence Global Observatory, you'd want to know ahead of time what's your clock.
Yeah.
I think the eDNA data is at more risk for something snooping us. Because you get an AI bot, it could go through, suck up all that sequence data with bare minimal metadata and analysis.
Yeah, I'll just say from the AS, you have a second analysis that would contribute for somebody to like to define and do based off of that. I know that there are people who already do that and like data sets generated from organisms to look at things like their content diversity. So very truthful.
Is it possible to just do like the raw sequence files if there's no information about barcode or anything? It's
Yeah, I mean, that certainly increases the cost of them attempting to draw meaningful inferences from it. But I
they
mean,
don't know what the barcodes are, just even to flip them out of them, what it means.
but I don't think that if you were trying to satisfy the requirements of the journal, I don't think submitting the raw data files within the corresponding.
These are the ones that are published yet, right? We should put that in the rest.
It comes down to, if you publish, it's gonna be in London. And you have no choice. And then I think the STP-chips, it would be harder for somebody to accumulate all that information and do a meta-analysis without the detailed knowledge that we have. But the eDNA is where it's a sequence, and you blast it against the GenBank, and it doesn't sequence.
And there's a lat long associated with it too. So you can lay in the lat long for any environmental data like set and
So I think the embargo is the way to go. And then you take the chances, right? You always do this with data on the drive. I know I've been contacted with data monitors about data sets we have on the drive. So but you're not taking the chance. So the GEN tells you you have to do it, just like student answer is going to tell you have to do it. So it's not about us deciding that we're taking a chance. But I think we could justify the Genome Canada, we put it there, but we've got data embargo for 2 or 3 years. Gives us a bit of time. But after that, yeah.
And just Genome Canada, I can't remember, specify when we have to sort of database it. Can,
yes.
Let's say that there's, if we have a little bit of flexibility and we're allowed up to 1 year an embargo, then we justify being slower. We justify the Genome Canada. Why would be if it's slower?
Yeah.
Archiving it.
Yeah, Genome Canada has very specific guidelines on that. In the proposal, we pushed that and nobody said anything. I remember discussing it with Sarah and okay, let's say we can't, it's supposed to be X number of months after the data is generated. Like, so it's the middle of the grant, it's supposed to be done before, and we said, no, it's gonna, the clock's gonna start ticking again. I think that's what we put in. Then we said the proposal, so, but I think you're gonna do the final report if we say sort of data legacy, so that, you know, everything will be on publicly available database. However, we're going to do data embargoes so that we can complete our analysis up to 3 years.
It'll be hard for us to get the money back if they don't like set up to get the data out there. Medium to short and medium timeline. Right now, the GEN-FISH domain is suspended for 3 years, but the GEN-FISH OneDrive area, I'm under a lot of pressure to get that basically called down to some absurd amount by September 1st. Now, the reason I asked the program zone is if there's some influence from ORIS or from the VP of Research Office to, you know, basically justify this Microsoft space, that might help us out.
Yeah.
Because now it's just not us, it's not just GEN-FISH project, it's something that has some living, breathing to it that moves forward.
And I think that's sort of one of the things we're gonna have to deal with as we wrap up GEN-FISH. And I think that we could get Trevor, probably the research to support that. not indefinitely.
Oh,
So we might get a year or two saying, okay.
But still, we can get it on these huge mainframe.
Yeah.
Yes,
Last month.
yes.
And that's, you Windsor has been really supportive. I would say, yeah. And it's.
They had a very $30 deficit, and now there's.
Yes.
And Microsoft changed licensing structure.
Yes.
So now they're absolutely nailed.
Yeah.
We can use the overhead, the grant folder. To help support.
Tons of it.
Just, yeah, like the sad thing is, you don't get it, is given.
That's much, you know. That much.
It's just a little prestige.
Dan, would you mind wrapping these up?
Sure. So, it's gonna be short.
Yeah, so I've found these 2 days really valuable. And they've been valuable in that it's reminded me what we've done. And that's very impressive. It's encouraged me that there's more to be done and that there's an appetite for that and there's costs and benefits. But in fact, I think moving forward mostly sounds like it's going to be beneficial for all involved. So I don't think this is going to be July 1st, drop tools and walk away from it. I think we'll have a really living legacy. Not all PIs for sure, but certainly a key group of PIs I think are going to continue working on this concepts simply because it's interesting and it's cool. And then the nuts and bolts of shutting down a large project never occurred to me until this, well, it actually has come home to roost just at this meeting. And that's I'm not going to be doing another big project, but it's a lesson hopefully lots of PIs take is don't forget the winding down time and energy costs because it's substantial. And, I've got an amazing team. We've got an amazing team supporting us for GEN-FISH, and they're going to continue to work for now a month and a half after we're done with GEN-FISH. And that's what's going to give us a good visual with Genome Canada, should anybody decide to move forward with Genome Canada. And the last thing is, it reminds me of the best part of GEN-FISH is such a cool group of people. The socializing with you all was a lot of fun and getting together. And somebody mentioned we should have a GEN-FISH reunion. I like that idea. We have to go for one of those IP grants. Just get together to, I don't know, ride boats and drink beer. See what happens. But does anybody want to say anything in conclusion? Because that's all I have to say for the wrap up. Nicola.
You deserve an amazing amount of praise for coming up with the idea and for leading us through this. Thank you.
Thank you very much. It was not always a pleasure, but there were always shining moments where I go, wow, that's so cool. And I think almost all of us learned from each other. So it did everything it should have. And I guess the last line is that in the first year of GEN-FISH, Sarah Jameson passed away. And Sarah was the person who kept me going on developing first the Discovery Accelerator and then the GEN-FISH. Lots of PIs contributed, but Sarah was there. He said, section 17.1.3.7 needs work and I want it by tonight. And that's how it got done and got in on time and got funded. So, projects don't get acknowledgements, but if there was one, lots of people be on there, but I dedicate the project to Sarah. So thanks very much, everybody. And we don't have that much longer in GEN-FISH, so. That'd be great.
Dating my user.